Hello, hello, testing, testing.
Okay. Good morning, everyone. No.
So, good morning, everyone. Welcome to the IMERC thesis presentation, the first session. I would just like quickly to introduce myself and the other members of the jury. I'm Karim Razini from the University of Algae. To my right is Audrey Geffen from the University of Bergen, and Camilla Roberta from University Polytechnic Maastricht, and Abed from the University of Gothenburg in Sweden. So you have a 15-minute presentation, and then we have 15 minutes of questions from the members of the jury, plus also people who are following online can ask questions, and also finally anybody in the room can ask questions. So about 2 minutes before your 15 minutes, I will just indicate if you're
2 minutes.
I'll just wave my hand at you. Okay.
Okay.
So start whenever you want.
Okay. Good morning, everyone. I'm honored to stand here today to present my master thesis entitled Ecophysiological Responses of Protoceratium reticulatum, an important harmful algae bloom forming species in Chile. This work has been possible thanks to my supervisor, the Professor Dr. Daniel Varela, and to my promoter, the Professor Dr. Ulrich Reichland. I will start by introducing you to the harmful algae bloom event subjects. They are proliferations of macro or microalgae with negative socioeconomic impacts on public health, coastal resources, and coastal commodities and fisheries. There has been a worldwide increase in harmful algae bloom, HABs, mainly due to the influence of climate change and the exploitation of coastal resources. Chile has followed this blooming increased trend, and the regions most affected by these events are within to the south, Los Lagos, Aysén, and Magallanes, which constitute the Chilean fjord. Chile is an important aquaculture country. It is the second world producer of mussels and salmon, and most of the production of these products takes place within Los Lagos region. Several microalgae species forming harmful algae bloom have been deeply studied because of the catastrophic event that they are responsible of. For example, in Chile in 2016, these two species produced the so-called Godzilla red tide, resulting in the largest fish farm and shellfish mortality ever recorded, equivalent to export losses of millions of dollars. However, there are still under-studied species, and this is the case of the dinoflagellate Protoceratium reticulatum. As you can see, it is dinoflagellate that is present in many parts of the world. It has a high tolerance to salinity, and blooms of this species have been detected only in temperate areas. In Chile, its optimal growth rate is between 16 and 18 degrees Celsius. Understanding the ecology of Protoceratium reticulatum is crucial to anticipate to future blooms. So far, what do we know about this species? Well, we know that it produces geotoxin, which has been related with invertebrate mass mortality events. Also, this formation allowed this species to alternate between the plankton and the benthos, and it has also been reported the allelopathic potential of Protoceratium reticulatum, which is the capacity to inhibit the growth of other species. I will now present the case that led to the development of my thesis, which is the case of Ilke Bay. In 2022, simultaneous closer levels of geotoxin, about those that are allowed by the regulations, were observed along the Reloncaví Sound. And those values high were also observed outside the summer season, when the conditions are not optimal for the Protoceratium reticulatum growth, which leads to think that the geotoxin was due to this resuspension and germination. The Reloncaví Sound is an important larval seabed for the mussel crops that are located at the end of the sound in the Bay of Ilke. Here, Protoceratium is presented as a threat to the shellfish industry. This is the Bay of Ilke with the mussel crops. Based on this problematic, our work had the following objectives. First, the characterization of the water environmental conditions that favor the development of Protoceratium reticulatum bloom and the accumulation of cyst in the sediment of the Bay of Ilke. Second, the estimation of the best temperature for the cyst germination. And third, the evaluation of allelopathy as a mechanism by which Protoceratium reticulatum can interact with other co-occurring species in the community. To do so, we sampled the water column environmental conditions during the austral summer season of 2023 in January, February, and March. Specifically, with a CTG, we measured temperature, salinity, dissolved oxygen, and chlorophyll A. We collected water samples from the surface and at CTG's depth. We identified the phytoplankton that was at that samples following the outer mold method, which involves fixation and sedimentation. Also in February, we sampled sediment at 10 sampling stations, and we measured the water column profile at those stations and the pH and redox potential of the sediment. With scuba diving, we placed 2 sediment traps, it made with 2 PVC tubes at 2 points of the bay, 2 stations, station 1 and station 10. And after 7 weeks, we came back to remove the sediment trap, and we collected more sediment at those stations, station 1 and station 10. These were extracted from the sediment by creating a density gradient with a fluid that is named Ludox. With the quantification of the cyst, we got an idea of the cyst distribution, with the cyst quantification over a time interval of the cyst loss from the sediment, and with the cyst containing the sediment trap of the cyst sedimentation rate. This is a picture of the variability of the cyst of different Dinoflagellates species that we observed, and these 2 are from Protoceratium reticulatum. Then, for the cyst germination experiment, we extracted cysts from sediments collected 3 years ago, not from cysts from this year, because Protoceratium reticulatum cysts have an obligatory dormancy period of at least 4 months. So we wanted to make sure that cysts were not in the dormancy. We placed 15 cysts per multi-well plate, and we incubated the plates at 3 different temperatures, 9, 15, and 21 degrees Celsius, 3 plates per treatment, and we monitored the germination success. Then for the allelopathy experiments, first we conducted a physical experiment which consisted of the co-culture of 2 species, Protoceratium reticulatum and another co-current and bloom-forming species, Heterosigma acacigo. Once we determined that Protoceratium reticulatum inhibited the growth of Heterosigma acacigo, we performed the chemical experiments to determine if that inhibition was due to the release of chemical compounds in the media. And to do so, we filtered the media of the cultures, and we inoculated new Heterosigma acacigo cells. We also renewed the media and the metals. And we tried on a dose-dependent experiment 3 different concentrations of the cell-free filtrates at 150 and 25%. So let's see what we found. Starting with the results from the field, the phytoplankton community was largely dominated by diatoms and followed by dinoflagellates, as has been shown previously in studies in the Patagonian fjords. This graph shows the dinoflagellate community at CHD's depth, and this is because Protoceratium reticulatum was absent at all the samples from the surface. It was also absent at CHD's depth during March. In January, it represented less than the 2% of the community, and in February, almost at 10%. It was in February when it reached its maximum density, which was 350 cells per liter. However, this concentration is far away from that has been reported in blooms of Protoceratium reticulatum during the austral summer and in the Chilean fjords. So with our results, we can say that we did not detect a bloom of Protoceratium reticulatum in the Bay of Ilke in the austral summer of 2023. If we take a look at the environmental conditions, during January and February, we can observe the maximum stratification pattern and also the maximum surface temperature. In February, there was a slightly increase in salinity with respect to January that matches with the increase in the concentration of Protoceratium reticulatum cells. And in March, the stratification pattern was completely lost, the salinity increased, and the temperature decreased. This stratification pattern is typical from the fjords, the Chilean fjords, because at the beginning of the austral summer is when they receive the most freshwater input coming from river discharge and ice melt, and the Bay of Ilke receives water from the Rilongari Sound. Water temperature was optimal for the growth of Protoceratium reticulatum during January and February, but what about salinity? It has been observed that blooms of Protoceratium reticulatum are associated with high salinity, and also they are associated when higher blooms occurred when the El Niño Southern Oscillation conditions and the Southern Annular Mode positive index converge. When these two large-scale phenomena occurred at the same time, there is a weakening in the vertical stratification and the advection of saline-rich nutrient water. During our study, the SAM index was positive, but the ENSO conditions were neutral. Therefore, our study serves to provide new evidence that the El Niño Southern Oscillation, together with SAM positive index, might trigger the development of Protoceratium reticulatum blooms. Cysts were found in all of the stations, ranging from 50 to almost 400 cysts per milliliter. The average cyst density was higher than what has been reported also in the Bay of Ilke in 2016. And also it was higher than Protoceratium reticulatum cyst observed in other studies in southern Chile. We observed significant differences on the environmental conditions among sampling stations, but the cyst density was not correlated with any of the environmental conditions or with depth. Therefore, although we couldn't establish a relationship between the cyst density and the environmental conditions, with our results, we can say that the Bay of Ilke is a potential cyst bank where blooms are likely to occur as soon as the conditions are optimal. Regarding the sediment traps at the station 10 were broken by the current, so we just had the data from the sediment traps at the station 1. At that station, the cyst loss from the sediment was higher than the cyst sedimentation, suggesting that the accumulated cyst in the sediment would gradually decrease over time, but however, more evidence is necessary to extrapolate this result to the entire bay and to other seasons. We obtained an extremely low germination success, just 3 out of 130 cysts germinated. One of them at 9 degrees Celsius and 2 at 15. This is one of the merged cells, and the remaining is covered with a tiny opening. So, as mentioned before, it's unlikely that the cells were still in the dormancy. So, one might think that we were missing any key factor triggering the germination, for example, the mechanical disturbance. But Salgado and collaborators obtained 80% of germination success at 15 degrees, doing exactly the same protocol as we did. So, although dinoflagellate cysts can survive in the sediment for decades, it is possible that cysts from Protoceratium reticulatum have a best before germination date, after which their viability decreases. And finally, the allelopathy experiments. The co-cultures of the physical experiment significantly inhibit the growth of Heterosigma acaciua. The inhibition was very strong because it was observed since the first day, and at day 8, all the cells of Heterosigma acaciua were dead. This inhibition has also been observed in a previous study where authors exposed Heterosigma acaciua to another dinoflagellate, Alexandrium minutum. Heterosigma acaciua species are also known for their allelopathic effect, but we did not observe any effect on Protoceratium reticulatum growth. With the chemical experiment, we confirmed that the effect observed in the physical experiment was due to the release of chemical substances, because the co-culture filtrate here in red was, when Heterosigma acaciua was exposed to the co-culture filtrate, its growth was significantly reduced with respect to the control. And we did not observe a significant effect in the monoculture, suggesting that the release of chemicals, allelopathic compounds, takes place only when both species are present. And finally, in the dose-dependent experiment, only the 100% of the concentration, here in red, inhibits significantly the growth with respect to the control. And also here in the dose experiment, as we inoculated different initial concentration to accelerate the allelopathic effect, we repeated the co-culture treatment. as in the physical experiment. But here we did not observe an effect on Heterosigma cashiwo when it was co-cultured with Protoceratium reticulatum. The competitive outcome can be affected by starting cell concentration, as has been shown previously. So from our results, we can conclude that the allelopathic effect is dependent on cell concentration. To conclude, the present study first identified a Protoceratium reticulatum cyst bank in the sediments of the Bay of Ilke during the austral summer of 2023. Second, provides new evidence to support the hypothesis that blooms of Protoceratium reticulatum are likely to occur during years characterized by a combination of El Niño southern oscillation and southern positive annular mode. Third, suggests that Protoceratium reticulatum cyst viability might decrease over time. And fourth, reported for the first time the allelopathic potential of Protoceratium reticulatum to negatively influence the growth of Heterosigma cashiwo, revealing that allelopathy is likely to be one of the mechanisms that favor Protoceratium reticulatum over other species to form bloom. However, further research is needed to continue monitoring the evolution of the identified cyst bank in the Bay of Ilke, and to disentangle the environmental conditions that influence the bloom formation, the cyst sedimentation, the cyst loss from the sediment, and the cyst germination. So thank you very much for your attention. Here are my references.
Thank you very much. Perfect timing, just inside 15 minutes. So now I ask the members of the jury if they have any questions. Who would like to start?
I can start. The allopathetic compounds, do you know anything about them? Do you know what kind of chemicals can be?
So in the case of Heterosigma cashiwo, it's ROS species, oxygen-reducing species, and also some acids, such as the oxalic acid. And the compounds of Protoceratium reticulatum haven't been identified because the allelopathic potential was just confirmed when it was co-cultured with Rhodomonas. So they haven't been identified, but as it is a dinoflagellated species, it's likely that it also has oxalic acid. And another of our objectives, but we didn't have time, was to analyze the allelopathic compounds, like to at least to extract the fraction in which they had the potential. Yes.
What do you think affects the viability of the cysts in the sediment? What do you think about the sediment conditions that might affect them?
So it has been seen that anoxic conditions preserve the cyst because it's not clear what are exactly the environmental factors that make cysts to germinate. But it is thought that conditions similar to those optimal for the cell development are also the optimal that make the cysts to germinate. So, for example, anoxic conditions avoid the germination, and, for example, a sediment with a pH and reducing redox potential, and, for example, with a finer sediment sand, those conditions are appropriate to preserve the cyst.
What do you think you could have changed from the protocol that you used for the germination? That if something you could have changed to make the germination better in the experiment, or is just because it was a too long period that the sediment was collected, and thus the germination was not optimal?
So one of the things that I have my doubt if it worked was preserve the sediments, because in the sediments they also have to be stored preserving those anoxic conditions. So after 3 years, it's possible that the anoxic conditions of the jar where the cyst with the sediment, maybe those conditions were lost. But what was in my hand, maybe it I could have agitated the multi-well plates to simulate the mechanical disturbance that the fish have when they are in the sediment and suddenly they found themselves in the water column. But I didn't have the agitator, the mechanical agitator.
The temperature range that you used for the germination, is that it was quite wide. Is that the normal temperature range for that area over the year? It was like from 5 to, I don't know, quite a wide temperature range.
So 15 was the optimal temperature, almost in the optimal range for Protoceratium reticulatum growth, and it's also the average temperature of the sea in summer. And then the 21 degrees were the predictable temperature variable of the EPCC. And then the 9 degrees is because it's the temperature that the fjord can reach during the winter. So as we observed that the values in the Reloncavi Sound were very high, the gestotoxin that I presented at the beginning in the introduction were very high, and those values maybe were due to a resuspension of cyst. We wanted to know if cyst can't germinate during the winter. So because of that, the 9 degrees.
Anybody from the audience wants to make a question?
Can I ask in general about the historical patterns of the red tides? And do you know what the critical density is for the cells, the critical abundance or the critical density for these species? Up until at what point? What's the threshold before they turn toxic, or they start exuding toxins?
Well, it has been shown that different cells can produce different concentration of gestotoxin, and also it depends on the strain. But when in Chile, blooms of Protoceratium reticulatum have been toxic, cells were 22,000 cells per liter. So that's the reference value that I use to compare them with the cells that I observed. Yes.
Can I ask, what is the purpose, or what was the purpose of collecting water samples at the second disc depth as opposed to like 5 meters or like the standardized?
So, Protoceratium reticulatum mainly lies within 8 meters, but it varies its distribution depending on the stratification pattern, because it has a high tolerance to salinity and it is favored by the high salinity. So we didn't know exactly in which layer it was going to be, and we didn't have like many resources to do a lot of samples that then I could analyze. So we took samples at the surface and then set this depth, because it is the depth where the light, the visibility, you throw a disc, and when you don't see the disc anymore, that's the Secchidis depth. So yes, and also because we use also this project with a parallel project that was comparing hyperspectral images of the water with the cells contained in the water.
And see, we don't have any online questions in the chat. We still have a couple of minutes. If anybody else wants to make any questions.
I'll throw one out there.
Okay.
Take one for the fish farms. So, why do you think these have been linked to aquaculture, the increase in red tides?
Well, mainly because aquaculture releases many nutrients into the water. So I think that it also explains why harmful algae bloom have been increasing over the last years in Chile. Because I could see with my eyes, like in the middle of a super pristine fjord, a salmon farm. So, and then you can also see like nutrients in the water, like dirty water around. So I think that favors the development of blooms.
But you could make the same case for sewage outfall from towns or runoff from farms?
Yes,
Does that lead to red tides?
I think so because specifically in the Bay of Ilke, it also receives input from the agronomical activities, and that river discharge that brings water into the bay, it also brings the water that has been collecting nutrients and herbicides and everything. So I think it also has a coastal land input. Thank you.
Okay.
Hold on.
Yes, well, I think we have to move on to the next one. Thank you very
Okay,
much.
thank you very much.
Okay, so the next speaker is Sharif Abdullah.
Yeah.
Please, you have 15 minutes.
Good morning, everyone. I'm Sharif Abdullah. I'm going to present to you impact assessment of non-mechanical harvesting of Ascophyllum nodosum on the regeneration of Vertebrata lanusa and its associated biodiversity. I think that's the. There's something wrong with the pictures. The pictures are not loading. No, this is the wrong. This is the wrong presentation. Wrong presentation of the program. I uploaded the wrong file, but I'm sorry about that. I uploaded the wrong file. Okay, so I'm going to present today the impact assessment of non-mechanical harvesting of Ascophyllum nodosum on the regeneration of Vertebrata lanusa and its associated biodiversity. So the wild harvesting of commercial harvesting of seaweed of the Ascophyllum nodosum occurs on both sides of the North Atlantic, and it's mainly due to applications such as fertilizers, medicinal and cosmetic products, but also animal feeds. In the west of Ireland, especially in counties like Galway, Donegal, and Mayo, they are harvested. Around 5,000 to 6,000 tons per year are harvested, thus creating revenues and jobs for the local communities over there. There are two methods of harvesting the seaweed. The first one is by hand, like basically going on the low tide and cutting the seaweed using sickles and hand, and it's the traditional method. The exploitation rate is 70%. And the other method is harvesting by boat through racks, and these are the racks. It was basically introduced in Canada, and the racks, they target to cut the seaweed above the holdfast, which is responsible for the regeneration of the seaweed, has an exploitation rate of 30%. So in theory, harvesting by boat is more sustainable than harvesting by hand. The vertebrata lanusa is an epiphyte that grows on top of the Ascophyllum nodosum, and that's the vertebrata lanusa, the brown filamentous one, and it is less persistent towards grazing as compared to Ascophyllum nodosum, thus creating a habitat for the macroinvertebrates. So the objectives of my thesis is mainly assess the recovery of the vertebrata lanusa and also its associated biodiversity after the non-mechanical harvesting in Kinvara Bay in Galway Bay Island. The hypothesis is mainly the boat harvesting has a less impact on the vertebrata lanusa biomass and its associated biodiversity than the hand harvest. For the sampling area was conducted in the Kinvara Bay, and that's located in Galway, in the western Ireland. There was 4 intertidal surveys were conducted. The first one was a baseline survey before any harvesting occurred. That was in July 2020, and then the company that harvests seaweed went and harvest the experimental plots because we want to understand what's going on in the industry, and that happened in September 2020. 3 post-harvest surveys occurred after that. The S1, which was 1 month post-harvest, and then there was a 6-month post-harvest, and then there was another 1 year post-harvest. Just to understand the recovery, the impact of the harvest and the recovery. For the experimental plot, I'm not sure if you can see it over here. There is a blue rope, like it was a 25 by 25 propylene, using propylene rope. And then there were 3 plots. The first one is the hand plot. The second one is the bow plot. The third one is the control, which was untouched. The sampling was mainly by random sampling. 6 replicates were per treatment level, like for each one, the hand harvest, the boat harvest, and the control harvest. The samples were collected in plastic bags. And then after we went to the lab, they separated the Ascophyllum nodosum from the vertebrata lanusa, and they stored it in industrial methylated spirit, which is IMS. My work started from here, so I took the samples, washed them in a sieve, and then separated the macroinvertebrates. It took me 4 months to do that. And then I dried the seaweed and measured its biomass and measured its dry weight. After that, I identified all individuals to the species level if possible, some of them to the genus level, and then checked their names on WRS, the World Registry for Marine Species. So first, to detect the effect of the harvesting and the recovery on the seaweed. On the x-axis, you have the different treatment, the control, the boat, and the hand. The y-axis, the dry biomass. There was a significant decrease from the baseline to the 1 month post-harvest, and then it was more or less, there was an increase or more or less the same. And that was unexpected. So when we inspected the images for the quadrant was mainly, it had a lot of fucus, and the vertebrata lanusa is an epiphyte that prefers Ascophyllum more than the fucus. So that's why there was a decrease. For the boat harvest, it was as expected. It started with the baseline. There was a decrease followed by increase. 1 month post-harvest, followed by an increase 6 months and then 1 year. It was kind of the same. For the hand harvest, there was a decrease in the average, followed by an unexpected increase, and then followed by a significant decrease. When we inspected the plot over here of the quadrant, we found out, I don't know if you can see that. So this Ascophyllum, it's formed of thick filaments. It has never been harvested. So even though the company would go to the experimental plot, the 25 by 25 has a 70% exploitation rate. So some areas were left untouched. So that when the plot landed randomly, it collected Ascophyllum that has never been harvested. And as you find here on the upper left, there is a thin filaments of Ascophyllum, and that's a regenerated one, but it's a small portion of it. So, and then like for the biodiversity analysis, the same on the x-axis. On the y-axis, you have the channel index. The control followed the biomass, the algal biomass. So there was a decrease 1 month post-harvest, and then a decrease in the average, and then an increase in the average 1 year post-harvest. For the boat sample, there was kind of constant until it dipped after 1 year. For the hand harvest, there was a decrease in the average, and it continued to decrease, and then slight increase after 1 year post-harvest. But more or less, they are comparable, the boat to the hand harvest, in terms of the channel index. I did a spearmint correlation to understand and then found out that the Shannon-Wheeler index is more correlated to PILO index more than the species richness, which like shows that biodiversity can be explained by evenness more than species richness. And then so like I plotted the K-dominance plot to understand the evenness of the community. The first section is the control samples. So it was more or less even, except for the 6 month post-harvest was slightly elevated, and then it was more or less similar. For the hand harvest, started with an even community, and then you find like the curve is elevated, which shows it is less even. It stayed the same 6 month post-harvest, and then it became more even 1 year post-harvest. For the both samples, you would find here that the community started that it was even, more or less the same, 1 month post-harvest, 6 month post-harvest, and then 1 year slightly elevated upwards. So I tried to inspect like what's happening. So I plotted the top 10 abundant species with their relative abundance. Here you have the baseline S1, S2, S3. Most of the community here, regardless of which phase of the control samples, is basically dominated by crustaceans that are grazers, like Hayal, Dixamine, Afarusa, Gyra. I went here with the boat treatment. It started with a community that's dominated by crustaceans. After 1 month of post-harvest, there was an increase in Perengia Pusillina, and that's like a detritus species. And then there was an increase, and then in S2, there was like a little bit, the community was dominated by Littorina, which is a grazer, but also 2 detritus species, which is Sickinia circuloides and Deculeps nitens. And then the community showed healing or like, because it was dominated again by crustaceans after 1 year post-harvest. So for the hand harvest, you would find here that most of the community is dominated by crustaceans for the baseline. 1 month post-harvesting by hand, there was a huge increase, 53% of Perengia Pusillina, which is a detritus species. And then 6 months post-harvest, that was the sample that I showed you the problem with, that the quadrat landed on a seaweed that has never been harvested. So it's mostly dominated by crustaceans. But then 1 year post-harvest, you still find around 35% is also dominated by Perengia Pusillina and Pusillina species, which is also detritus. So like the both work better in a way. I did a correlation analysis between the vertebrata and biomass, and it showed there was moderately negatively correlated to PILO and moderately positively correlated with species richness. For the NMDS plots, there was a clear separation between the baseline and S1 and S2 and S3. So baseline and S1, these were the samples were analyzed by a student in 2020. So we kind of speculate, or we think that it's because of inconsistency in identification. The stress value is 0.16. There's not much to say here, but however, here, these are the samples that I analyzed. You would find that so I don't know if you can see, but in the blue squares, these are the hand harvest samples. S2, so 6-month post-harvesting, and the controlled post-harvesting. They are mostly below 1 year post-harvesting samples. The only samples that are near the S3, the 1 year post-harvest, is the boat from 6-month post-harvesting, showing that the community is kind of healed, or it's doing better than those that harvested by hand. So I just want to, for the conclusion, samples from the boat harvest plot seems to show signs of recovery in terms of biodiversity after 6 months compared to hand harvest plots. It's a challenge to judge the recovery of the Villanosa biomass from my data. There was a problem with the taxonomic issue, and my supervisor is intending to fix like the identification of the previous student. And we should include another site to also see if we would see the same pattern, and the boat might work better than the hand harvest. Yeah. And like, I just want to thank my supervisor and my friends for helping me throughout this journey. Thank you so much.
Questions?
I can start.
Yeah, please.
Well, Ascophyllum is a slow-growing species, so the regrowth after harvesting is really slow. But you did have the Protanosa coming back. So, do you think it's growing on like fronts that are not harvested, or is it growing like further down on the Ascophyllum? Because usually the Protanosa grows in the top part of the front.
So to be honest, I don't know, because I just I analyzed Villanosa stored in IMS.
Yeah.
Like I didn't see it in the field. I don't have I didn't inspect this in the field. Like, to be honest.
Why do you think you observe these changes in the community associated from crustacean to gastropod, and again from crustacean in some cases?
So like what happened is that when they harvest the seaweed, there was a niche. So like just the detritus, I don't know, maybe things died, so the detritus grew and it dominated the community.
And I have another question. Do you have an idea of why you found a negative correlation between the algal biomass and the diversity, the Pilo index?
So the Pilo, it's like measured the evenness of the community, and like starting from the baseline, it's mostly dominated by crustaceans, and then when it hits like the harvesting happens, it the community becomes more even, because like some species are hit and then it leaves a niche to species to grow, so it become more even. So whenever you cut or harvest the biomass, the evenness increase, and that's like my speculation. I just want to also know that I didn't submit my manuscript, and I'm in a stage that the plots are finalized, but the interpretation is not finalized. So this is my interpretation. I haven't sat with my supervisor and discussed thoroughly about the interpretation.
Do you know anything about the recruitment patterns of some of these colonizing species? There's seasonality, or
Unfortunately, I didn't dig in depth in the interpretation, so I also need to know their functional diversity, and to know like a lot about species in specific, but I didn't have time to do that.
oh, just curious.
I just received my visa very late for Ireland and North Schengen, so I came in February, started very late.
And you use mainly non-parametric statistics. I was wondering why,
Because
in some cases, why you could have used, like instead of Permanova, why did you use Permanova and not?
So I checked, like I first started with like, so I first checked in there if they abide by being normally distributed, and it didn't meet the conditions. I tried to do log standardization, also didn't meet the conditions, so that's why I finally went to parametric analysis. Yeah.
Yeah. So, what's your conclusion then when it comes to harvesting? Maybe you mentioned it, but let's say, which type of harvesting do you prefer when it comes to the growth and the effect on the ecosystem?
So I do prefer like harvesting by boat because it's 30% exploitation rate, and also the racks are designed to cut above the hold fast. That's responsible for regeneration. That's in theory, but like you also want to make sure that the people who harvest has the experience, because the racks has to be sharp, they has to be bland so that they do not cut the whole plant, and they have to be a little bit experienced about it. As compared to the hand harvest, they are already experienced with it. So that's
Any questions from the audience?
yeah.
You mentioned that the rigs were used more in Canada. Are they also starting to be used more in Ireland as
Yeah,
well?
so like they're trying to apply it in Ireland by this specific company, and like they just want to see. So sustainability in the seaweed harvesting is the permits are given from the view of the algal biomass. So like they also the research lab wants also to see if it is sustainable in terms of the biodiversity of the community.
Cool, so it's like helping
Yeah,
inform
and
national
yeah, and
policy.
also like this project, it concentrates on the associated biodiversity to the Villanusa, but there are other people working on the associated biodiversity with the whole canopy, and also with the biodiversity within the sediments beneath the Ascough Island. So it's like trying to see it from all angles. Yes.
So did I understand what you said, that you were going to do the functional diversity, but you haven't got to it, or?
The interpretation, like I just like see each and every species, their function by diversity, like just try to inspect closer. Yeah, like the interpretation in general, like I need to spend more time on it because like I just.
Do you have a vague impression now, a preliminary impression now, whether grazers versus predators versus whatever?
Yeah, the preliminary most crustaceans that I searched for, they were grazers, like herbivores. But then when I searched for the gastropods that took that grew after harvesting, they were mostly detritivous. So that's why I expect that there's a negative impact on the community, and then the boat healed while the hand is still as it is.
We still have time for questions.
Yeah, why not? Can you go back to the MDS?
Sure.
Can you repeat again what you said about the boat? It's very difficult to actually see.
Yeah, I think
So
also
can
like
you-
in the screen, it's like the colors are better, but
Can you use the cursor?
sure.
Maybe
Sure.
to show us again which are the different dots and what they mean.
Okay, so like the triangles are the controls, the squares are the hand harvest, and the circles are the boat harvest. The blue is from S2, okay, and the purple is from S3, which is 1 year post-harvest. Again, you have circles, triangles the control, and the hand is the squares. So what I'm saying is that here you have S2, and then the blue were near the S3, which was harvested by boat. Okay? Yeah, that's why I'm saying that it might have the community from boat harvest healed or regenerated faster, because it was near the S3 as compared to here. But someone might argue that the control samples are clustering together with the hand harvest, and that why that? Because the plots in general, if you visit the field, they chose that the hand harvest is and the control are mainly formed of boulders, like rocks, but the boat plot, it doesn't have as much rocks because the boat would come at the high tide So yeah, and like I did also, I don't know if I have time, but I did also like a PCA, and then you have the control and the hand, they are projected together as compared to the boat. Here is the baseline, and then the one month, 1 year post harvest, and then the S2 and S3 are together in the same quadrant. So. You're welcome.
Okay?
Yeah. Thanks a lot.
Is any questions? No. Okay, thank you very much.
Okay.
I can edit it up and move it to another. Here,
Okay, right.
So we will not start now because we need to start on the time
At 10?
people are expecting because
Okay.
people are following online.
Perfect.
Okay. Okay, so you have
Okay.
another 6 or 7 minutes.
Okay.
Can you use the time to move a microphone closer? Yeah.
The microphone,
Like
please.
this. Yeah, should be on the camera. See how it's
If I'm here presenting, am I on it? So we start in 7 minutes, right?
Yes.
She's got a video in her presentation, but it's not working in the presentation. We don't know why, but she has it in desktop. So maybe she's like turning off the presentation, turning on the video, and then she comes back, right?
Use up the time to show.
out of the frame here. Do you know if I'm on the frame here?
I look for something there.
Yeah,
Or
would be.
gonna be moving.
I can stay here. Thank you.
Ah, okay. Okay. No, you're right, you're right. Okay, so the next speaker is Suzanne Rugby, please, 15 minutes.
Okay. Hello everyone. So my name is Suzanne, and I'm going to present you my master thesis, which is named "Why Does Metabolic Rate Scale with Body Size?" So to start with, what is metabolic rate? Metabolic rate refers to the amount of energy that organisms spend a specific amount of time. We also call it the cost of living, because this is basically how much energy you spend to live, how much you spend to have your organs running, your body working, and so on. So it determines how organisms expend their energy and their need to consume resources, and can be measured through calorimetry, which is the heat production, or respirometry, which is the oxygen consumption. So I invite you to imagine an elephant, and next with this elephant, a tiny mouse. The elephant has an average mass of 4 tons for a metabolic rate of 40 kilograms per minute, 40 joules per kilogram per minute, while the mouse only has an average mass of 20 grams for a metabolic rate of 600 joules per kilogram per minute. So while the mouse is about 200,000 times lighter than the elephant, it still has a metabolic rate 15 times higher. Why does it scale out of proportion like this? It's because metabolic rate scales according to body mass, depending on this equation. Metabolic rate equals A times body mass exponent B. When we apply a logarithmic transformation to this equation to make it linear, as you can see here, we notice that the A here is the intercept of the line, while B is the slope. The scaling exponent B is the slope. So this is basically B that states how steep metabolic rate scales depending on body mass. This is because this value of B is lower than 1, that lighter animals, lighter organisms, have a higher metabolic rate. So B is very important because this is the scaling relationship between metabolic rate and body mass. But what is the value of B? We still don't know. It had been the topic of a century debate. As I was saying, for over a century, we believed that B was a constant and that its value was equal to 0.75. It had been established by the Claver's law in 1932. And so supporters of this theory believe that any deviation on this value of 0.75 are due to measurement errors. But more and more researchers actually believe that it might be wrong and that these deviations are not due to measurement errors, but that B might actually be a variable that varies depending on organisms and environments. Among the supporters of this new theory, that B is a variable, there is this guy. His name is Turbine. She's a researcher at the Technical University of Denmark, and she published last year a paper in Integrative and Comparative Biology called Growth and Mortality as Causes of Variation in Metabolic Scaling Among Tax and Taxonomic Levels. That's quite a complicated name, but he was basically demonstrated in this paper that we can find that there is a positive relationship between growth rate and the scaling exponent B, and that this relationship might be due to mortality rates occurring in brown trout and tuna. So from this observation, he built this hypothesis that maybe metabolic scaling is actually shaped by selection pressures occurring during early life stages. But as I told you, he did so in brown trout and tuna, which are both species with very high mortality rates in early life. So in the scope of my thesis, I wanted to verify if this hypothesis might also be true in species with low early life mortality rate. So to do so, I went in Morea, French Polynesia, to verify if this hypothesis might be true on other species. Before we go further, you need to understand three different things. In my thesis, I'm going to talk about three different taxonomic levels. First, the evolutionary scaling, which refers to the scaling, which compares the metabolic rate across species. Then the static scaling, which compares the mortality rate between different individuals of the same species at the same age. And last, the ontogenetic scaling, which compares metabolic scaling, which compares metabolic rate, along the life of the same individual. And then I measured three different metabolic traits. First, the standard metabolic rate, SMR, which is basically the energy you are spending when you are at rest. So when you are not doing anything, you still spend some energy, as I was saying, just to have your organs running. This is the standard metabolic rate, SMR. Then the maximum metabolic rate, which refers to the maximum energy you are spending when you are exercising high physical activity. This is MMR. And last, the aerobic scope, AS, which refers to the organism's capacity to increase its metabolic rate to support activity. And the aerobic scope is basically the difference between MMR, the maximum metabolic rate, and SMR, the standard metabolic rate. So, as I told you, I wanted to work on species with low early life mortality. It's quite hard to quantify early life mortality rates in fish, so I used as a proxy parental care. I assumed that species with a high level of parental care would be more likely to have low mortality rates. So I worked on these two species, the orange fin anemone fish, Amphiphorium chrysoterus, which is characterized by the male venting its eggs to provide them with oxygen, and the female defending them. And then I also worked with the striped cardinal fish, Osorhynchus angustatus, which is an apagonid, and as an apagonid, it's a mouth brooder. So the males are basically protecting their eggs in their mouth, just like you can see here. Once I found juveniles of these two species, I built a constant water flow system, providing water from the top, and I built some individual containers to keep track of who was who, to be able to do several measurements on the same individuals. And then I fed every individual in excess for a limited amount of time, for 1 hour, twice a day. Then I performed respirometry. I performed between 4 and 7 runs of respirometry on every individual, and I left an average of 1 week interval between every measurement so the fish could grow. I don't have time here to explain too much in detail how respirometry works, but I'd be happy to explain later. But basically, I had some glass chambers. The fish were in the glass chambers, and as the fish were breathing, I could see a decrease in the oxygen. This is what you can see in the curve here. And then every 2 minutes, this pump on the right was activated. It's a flush pump that was providing fish with normally oxygenated water, and so we could see the oxygen increasing again, as in this curve. The respirometry was measuring oxygenation every second, overday or overnight, so between 6 to 12 hours and 12 hours. And I also left a minimum fasting period of 16 hours prior to every respirometry measurement, so I was ensured to measure the standard metabolic rate and not digestion, since I wanted to measure the metabolic rate at rest. Unfortunately, this video was not working, but I also measured MMR through a chasing protocol as described by Kutt and al. In 2002. And to do so, I was chasing the fish during 60 seconds using a plastic pipette. Once the fish was getting tired and was exercising physical activity, I put it in the chamber, in the glass chamber, and measuring its metabolic rate. And after every respirometry measurement, I also weigh the fish to get the growth rate. So here are my results. I built linear mixed effect models to get ontogenetic and static scaling. You can see, it's quite small here, but you can see in the graph, on the y-axis, I have the SMR. I'm not going to present you the results for MMR and aerobic scope because of a lack of time here, but also because SMR had been the metabolic trait to be shown to be the most related to body mass, to body mass, and also because not all fish responded well to the chase. But so, yeah, the graph on the left, panel A, is for the Amphiprion chrysopterus, so the clownfish, and the graph on the right, panel B, refers to the apagonid, Ostoranchus angustatus. I have on the y-axis the measurement for SMR, and on the x-axis for body mass. The plain colorful traits represent the ontogenetic scaling, so this is basically the scaling for every individual fish, while the dashed black axis represents the static scaling. So this is the scaling across every individual of these species. So after building linear mixed effects models, I found no significant difference between the static and the ontogenetic scaling. This suggests that the static scaling exponent, so the value of B, closely matches the value of B, the static scaling exponent, so the value of B at the static level, closely matches the value of B at the ontogenetic level in species with low mortality rate. Then I studied the relationship between the scaling exponent of SMR, so the value of B, and growth rate. So to do so, in panel A and B, you see all the colorful lines. They all have different slopes. I used these slopes, so this is B, and I used this slope and I plotted them against the growth rate. This is what you can see in panel C and D. And then I conducted a Pearson correlation analysis to study their relationship, and I did find a positive relationship for the clownfish in panel C, but not for the apagonid in panel D. I was not so surprised of not finding any significant relationship for the apagonids, because I had very broad confidence intervals for these species, showing that these results were associated with some uncertainties, probably because I conducted the measurement for the apagonids for way shorter time than for the clownfish. For the apagonids, I had some respirometry lasting about 6 hours, while it was about 12 hours for the apagonids. But yeah, so I had very broad confidence interval, and by the way, in panel B, you can see that the ontogenetic scaling lines are quite spread out. But I did find a positive relationship between the scaling exponent for SMR and growth rate for the clownfish. This suggests that individuals that grow fast exhibit a steeper metabolic scaling at ontogenetic level. Steeper ontogenetic scaling means that the value of B is higher. So far demonstrated that individuals that grow fast, at least for the clownfish, tend to have a value of B being higher at the individual level, and also that the static scaling exponent and the ontogenetic scaling exponent are very close from each other in species with low early life mortality. As a reminder, the hypothesis was that metabolic scaling was shaped by selection pressures in early life. If this hypothesis is true, we can expect in species with high early life mortality to grow fast, to go out of this stage when they are highly vulnerable. And so if this is true, we would have an indirect selection for fast-growing species, for individuals with high early life mortality. So if we have this indirect selection for fast-growing species, we could expect a big difference between the ontogenetic and the static level for the scaling exponent, and so on between the static and evolutionary scaling exponents. But this is what I showed you here. But I didn't have species with high mortality rates, since I picked species with low parental care. So in my case, I was expecting not a big difference in the scaling exponent between ontogenic and static scaling. This is what I showed you in panel A and B. And then as for the difference between static and evolutionary scaling, it depends on what species we put in the scope of the evolutionary. In my data, I used species from the last paper published by Noreen, and they were mostly ray finfish, teleosts, that typically exhibit high early life mortality, according to Hood and Sogart. So if they are exhibiting high early life mortality, it means that we could expect a strong selection for fast growth rate, and then a high scaling exponent at the evolutionary level. This is what I found in my results. Between the ontogenetic and the static level, I did not find any significant relationship, but I found a very high scaling exponent at the evolutionary level, which matches the hypothesis. So these results suggest that selection on growth rates drives variation in metabolic scaling. So to conclude, my results tend to verify a new hypothesis that metabolic scaling is shaped by selection pressure in early life. And to do so, I used a unique approach by investigating the scaling pattern at 3 different taxonomic levels all at once, and I used species with high parental care. This study is part of a broader research that aims to compare metabolic rates of species following different metabolic reproductive strategies. So we have further research to be done on this, and maybe a paper to publish later. Here are my references, and thank you very much for your attention. If you have any questions,
Thank you so much. We went a little bit over time, not very much.
Okay, so about the methods here, you have the fish and you put them in some chambers and you stress them and you do the same thing with the same individuals for a number of times. How will that affect the growth rate of the fish? Do you think it will affect? Do you have any controls to compare your experimental fishes with something else?
Yeah, I was not expecting any difference in the growth rate. I was just leaving 1 week interval so the fish would have time to grow. But basically, I was stressing the fish, but for 60 seconds, and then putting the fish in some glass chambers, and they were staying there overnight. And so when we were studying the metabolic rate, we could see, like, usually when they were stressed, they were breathing more deeply, and then it got stabilized. So basically, I was not really studying them under stress, but like at their standard metabolic rate. So as I was conducting the respirometry during 6 or 12 hours, the standard metabolic rate was stabilized, so I don't think I could have any influence on the growth rate. I was just like letting them grow as they would have done.
So I'll pick up Pear's question. I think his question was the more long-term, the repeated handling over a 6-week period. Did you have any control over, or to see whether or not that handling treatment had any effect on your results?
Do you mean if by doing this measurement I was impacting the growth? I didn't, but I was studying metabolic scaling, so I could anyways expect I was expecting higher metabolic rate for fast-growing species, but if the fish were growing faster, it wouldn't be an issue in my case, because I was not studying if they were growing slower. Okay, I was not studying if they were like growing faster or slower. I was just studying if individuals that grow fast had a higher scaling metabolic scaling exponent. So it didn't really matter if they were growing slower, faster, or at the same level in my case.
Sorry, but it has been seen that stress can affect the actual final size of an organism. So, for example, temperature is known that affect the final size if juveniles, for example, larvae, are subjected to higher temperature, increasing temperature for the global climate change. So if you didn't have any control, to see without any stress how was actually growing, how can you say that? I don't know if I will explain myself.
I'll pick up what
Yeah,
exactly.
thank you.
Yeah.
Okay, so how do you know that your continued handling did not impact the fastest growers? And so that you in fact did not get the maximum growth rate that you could have seen within that group?
Yeah, that I don't know about that, but the thing is that I don't really mind if it was affecting their growth rate, because I was not studying the growth rate, but only the relationship between their growth rate and their metabolic rate. So
yes,
even
but if you impacted the growth rate of the fastest growers, you don't have those individuals expressing their full potential in a way.
okay,
You've narrowed the scope.
okay, I see.
Yeah.
Yeah, okay.
It's hard also for us to express that.
Yeah.
That's what
No, I see what you mean. Also, like I conducted when I said that I had a week interval between every measurement, it was more like a minimum, because I conducted this experiment over a few months, and I was doing like one overday or overnight measurement, and at the end, every individual had between 4 and 7 measurements. But I conducted the research over 4 months. So at the end, they were experiencing this stress only once. For sometimes it could be once a week. Sometimes it was like once a month. I had like, when I was getting new individuals, I was conducting respirometry in them, and so I didn't have the same interval between everyone. So it was quite randomized.
There are ways that you could actually compare to see whether it was happening. I have another question. So, a while ago, there was a lot of work on fish having friends, and especially larvae and early juveniles, that in fact their metabolic rate was lower when they swam with friends. And so my question to you is, when you were following especially the anemone fish, where you've got a cluster that hang out together for quite a long time, did you think that might have affected the way you saw them compared to, for example, the cardinal fish?
Mm.
Did you put them? Did you test broods together?
No.
As opposed to mixed?
The fish were kept in 2 individual containers, so they were having some small Tupperware boxes that I let those in, so they were not in contact with each other. But also, we collected the eggs of these fish, and then for the clownfish at least, we collected the eggs and then we reeled the larvae from the eggs. So they didn't really experience the social life.
So you didn't keep broods together; they were all mixed.
Yeah, all individual containers. As soon as they were starting to have strays.
Can you just go back to one with the slide that has the 6 figures and lots of figures? And, yeah, the top 2 slides, you have some individual lines are horizontal or even with a negative slope. What's the explanation of that?
Yeah, I used some coexistence to remove the outliers in some graphs, but sometimes they were not considered as outliers. I used the coexistence in this graph, but I believe that the horizontal or negative slopes or the outlier, here I removed an outlier, I think this is one of them. Like sometimes I just had some odd results that I couldn't really explain, but like maybe, I don't know, maybe something happened in the room, like the light was switched on and the fish was stressed, so it impacted the result. But yeah, I tried to remove the outliers from this stage, but from this stage it was harder to remove, like to identify how could I decide that I was removing this data and not another one.
I have one more question.
Yeah?
What is the mortality rate of these guys compared to, you said these are low mortality rates, so what's a high mortality rate and what's a low mortality rate?
Do you mean between my two species?
Between your two species and between your two species and, say, any other fish?
Yeah, so yeah, as I was saying, it's hard to quantify mortality rate, so I use as a proxy parental care. Of course, like we can't really say for sure that it's because you have high parental care that we have low mortality rates, but it's the best that I found. And I didn't have, here I wanted to study species with a low mortality rate, so high parental care, and they both have different level of parental care, but we can't really compare like which is the stronger one. They just have different behaviors.
What kind of mortalities did you get in your tanks?
In my tanks?
Yes.
At the larval stage, I had quite a lot of mortality. We always have a lot of mortality. I read some books, and apparently, yeah, we always have quite a lot of mortality at the larval stage, and I struggled a bit with them, to be honest. That's why I had less clownfish individuals that apagonids, because I got all the apagonids from the wild at the juvenile stage, while I reared the clownfish larvae from their eggs. So I did have some mortality, but same, it's very hard to quantify because when the eggs are spawning, you don't get always the same number, but sometimes you get like 30 larvae, sometimes 50, and it's impossible to count because they're just all swimming around in this aquarium. So.
But you followed individuals over a long period of time.
Do you mean in the, yeah?
It's all right, I think we're.
Okay.
Any questions from the audience?
Say something, there's a comment here in the chat by Dominorin. He tells us that she also compared her fish's growth rate with the growth rate of individuals from the same species from the wild, and that she found no significant difference. Just coming back to the growth rate we talked about before, that she found no differences between the growth rate in the wild and between the individuals and her ones. Just know that the care supervisor role not here.
There are no more questions? Yes?
Do you think the hypothesis you present here is a disagreement with the volunteers like Brown and West? And if you think that it is, do you have an alternative theory? Because metabolic scaling with the value for the exponent, which is still debated, is based on very fundamental principles of physiology and scaling of physiology.
So the theory established by Kleiber that states that the scaling exponent equals 0.75 had been based on the two different things. First, some empiric studies. A lot of papers found this value of 0.75 or some values very close, and they believed that there were deviation errors. And then it was also hypothetically verified by some fractal theory. For example, he believed that this value of 0.75 was the perfect value to explain like how small vessels were, like how big vessel could be spread out into smaller vessels, and that this value was explaining anything about physiology. But yeah, in this case, like after seeing so many values that deviate from 0.75, I actually believe that maybe they're not just mistakes or like mistakes, but that maybe metabolic scaling might be selected by selection pressures, and then that this is happening in early life, and then that maybe later in life the environment is having a stronger impact. Did I answer your question?
Okay, thank you very much, everybody. Yes, coffee break now?
Hi, good morning everyone. I'm Hayes, and today I'm going to introduce how I use a mechanistic model to look at the impact of fishing on the global biogeography of marine fish food web and the trade-off between management objectives. So traditional fishery management focus on single species assessment, but then sometimes they can overlook the interaction in the ecosystem caused by and impacted by fishing. And therefore, in recent years, EBFM, ecosystem-based fishery management, is gaining more popularity, and marine ecosystem model, the MEM here, become an important tool when we are trying to employ EBFM. And this is a flow of employing a marine ecosystem model. We need Earth system model input, and then we choose a marine ecosystem model, and then we get the biomass and yield estimation. The MEM lies in a spectrum from mechanistic to empirical. Mechanistic is more, we use equation, biological processes and principles to resolve the interaction, while for empirical, it's more like using real world data and to use like the catch data to derive the trophic level in the ecosystem. And here are some examples of the marine ecosystem on this spectrum. They can be a mix, or for example, for DBEM here on the empirical end, it will be more like they're using catch data and then comprising 100 species of interaction map. And then, but then for mechanistic, we have our model, Feisty, that we only have 3 fish groups. We look at the biological processes by equation. And this is our major objective, to answer the question how to fish and how much fishing is appropriate in a fisher scenario. And the Earth system model that we use is UFIG. The input to Feisty from UFIG is zooplankton mortality, detrital flux, and the surface and bottom temperature. And the Feisty setup that we use is basic 2. There is some other setup in Feisty, but then it can be more complicated or it can be really simple, so we choose the medium option, the basic 2. And this is the composition of our model. And there is no species specified in our model, but then we group them according to their size and their feeding frequency. We have 2 resources here, the zooplankton and the benthic resources, and 3 fish groups. I will introduce them one by one. And then the size spectrum here follow a logarithmic scale. The first fish group is the forest fish, F. They are plantiforous with smaller size. The maximum size is 250 grams, and they live in the photic zone. And the big pelagic, the blue one here, they are way bigger. The max can go to 125 kilo, and they also live in the photic zone. And demersal, they have the same size spectrum as the pelagic fish, but then they sometimes in their life stage, they feed on benthic resources more. And these 3 equations, so Feisty is basically a big model with a lot of equations, but then these 3 are the backbones of the model. As you can see, they all have the W here because it's a size-based model. So it is scaled with the weight. And clearance rate, consumption rate, and metabolism weight. From this weight, we can further derive other parameters in the model. But then just for the clearance weight, if we want to look at the food encounter weight, we will need one more parameter, which is the theta here, the predator-prey size preference. And it follows this distribution. It's a normal distribution of the prey weight. And then, because we just prefer, the predator would prefer a certain size of the prey. And after having all this information, we can define the fishing scenarios. And these are the 2 curves that 2 equations that we use to calculate the yield, and we select a straw selectivity curve. And then we created a matrix, and in that matrix, there is different combination of fishing mortality on the 3 fish group, and then we apply the whole matrix to these 10 log-scaled total fishing mortality, TF, and it ranges from 0.2 to 20. And at each point, we do the matrix, and then we choose the combination, like for example here, that gives the highest yield among all those combinations. And then we will use this concept to look at and define 3 reference points. First, maximization of catch, we call them max catch F. And then it's just the point that we give the highest yield. And sus F, the maximum is also basically max catch F, but then we stop the fishing whenever any fish group, their spawning stock biomass is falling below 20% of the original. And then max rent F is the economic one. So the money sign there, and we have the price and cost function here. And because we don't have the information from like all around the globe, we just employ these 2 reference points globally and in 4 eco-region. But this, we only do it in the North Sea. And going back to this objective again, and we add one more, because since now I introduced all and how we can use mechanistic model. And so we will also focus on looking into how the ecosystem interaction look like at all these reference points. So first result, the yield composition at the first 2 reference points. Before looking into that, we have to make sure the biomass projection of our model is similar to what it is in the previous model. And so we compare, because we made some modification of the setup, and also we use a different Earth system model because of the computation cost. So it is kind of similar, but that there is a slightly higher production predicted in the Southern Ocean region, but our supervisor and I think that's kind of acceptable. So we go on and apply the fishing mortality. And here is the graph. I know it's a lot of math, it's a bit confusing, but then I'll go through it one column by one column. So the row are the fish group, and then here is the fishing mortality, the middle one is the biomass, and then they combine, give the yield. And first we look at the fishing mortality from, this is the when it's at sus F, sustainability F, so it's lower fishing mortality, to the max catch F. You can see that the distribution is relatively similar, but then the intensity increased a lot. We go back again, look at it again. Yes, the distribution is similar. And then the biomass in the middle, it's difficult to see, but then if we look into pelagic fish specifically, they decrease a lot, decrease the most. So they drop by 45%, while the whole total biomass only decreased by 24%. And last but not least, the yield is mainly contribute, the increase of yield is, we can't see it that much in this calcium, but then it's mainly contributed by the forage fish. And then we look deeper into the 4 eco-region that I mentioned. This is the shelf region, and these are the biomass, SSB, and yield curve of each species. And if we look at the pelagic fish, we can see that the blue curve decrease a lot more than the other 2 species. And then if we look at demersal fish, it's kind of weird, but then like its biomass remain high, and we're not fishing them at all. And I'll address that later. But for forage fish, we can see from this composition curve, we can see that forage fish, and then also there's the curve shape resemble each other. We can tell that the forage fish account for the highest proportion of the total yield. And these are the other eco-region that I collapsed those graph into a bar graph. This is sustainability F, and then this is the max catch F. And you can see eutrophic and mesotrophic, they are the open ocean, and there's not much there. And shelf region and the slope region, they're kind of similar. The biomass distribution is similar, but then usually the abundance is just lower. And so apart from, so Y here is the yield. So apart from the sus F in shelf region, all the other yield you can see is just forage fish. So it sounds a bit ridiculous. Why? It's just forage fish. According to model, we should just fish on forage fish if we want to maximize our catch. And because they have the highest resilience, and then they have the short lifespan, so you can just keep harvesting them. But then it depends on what we want. To what extent we should harvest the forage fish. So here is the debate of the forage fish, because in the previous year, we did have collapse of forage fish stock, and therefore some countries adopt the extra precautionary approach. But then these papers listed here, I don't have time to go through them, but then they all suggest that actually harvesting the forage fish can reduce the competition of forage fish with the juvenile of demersal and pelagic, and in turn increasing their biomass. And also another interesting viewpoint, instead of just increase the harvest or decrease the harvest, the author here suggests a post-exploitation strategy, because if we look at the paleo data, for a 500-year timescale, there's inevitable and frequent collapse of the forage population. So maybe we should just harvest based on how they fluctuate. And then we try to look at the ecosystem interaction and look at the mystery of why we're not fishing the demersal at all. So in the beginning, I thought I have some problem with my model, but then I realized that if we look into the fishing matrix more carefully, we can see that if I try to increase the demersal fishing mortality, the pelagic fish decrease a lot. Like the decreased expenditure is really great. So it's actually because demersal fish and pelagic fish, they have the same size class, so they are a strong competitor of each other, while pelagic fish is a major predator of forage fish. So when forage fish is comprising a big portion of the yield, it is good that the model choose to keep demersal high, so to keep pelagic low and keep more forage fish in the system. And we show that Fishery is capable of demonstrating these ecological processes. And therefore, I also did a mini test to look at what will happen if we allocate fishing mortality just to one group, and to see what those authors suggested if that is will. So you can see in the one that I allocate fishing mortality only to forage fish, the pelagic fish across all size class, the x-axis here is the size classes, their biomass increased a lot, and then at the 5th size class, demersal fish biomass also increased. It's just a really simple test, but I'm just trying to see if Fishery have the ability to demonstrate this fine-scale ecological consequence, and the servant is applied, and it does demonstrate it. And last but not least, we have the max rent F. These have a similar structure of the U-curve I showed before. These are the max rents for each species. And we can see that demersal here actually gives the highest yield and highest rent at max rent F, the blue vertical line, despite its lowest original biomass. And when we compare it back to the U-curve before, here on the left, I don't know, left or right, but then that curve, you can see that these rectangles show a similar region in the graph, and then they look further apart, but because it's a logarithm, it's a log-scaled x-axis, they are actually quite close to one. They're both quite close to one, but then their composition is different, the composition of the catch. So going back to the initial question, which reference point, how much we should fish, what species we should fish. First, we look at if we just look at max catch F, it sounds really ridiculous because the U is just comprised of a lot of forage fish. But then in reality, there's fishery naturally attain this max catch F point, because in Africa, food is a major objective. So they just catch whatever they have, and they catch all the smaller size fish. And turns out that region, the symbiont fishery, have the highest yield among the region. And such F and max rent F, we compared them before, and I said they're kind of close to each other, and it echoes with the MUI concept that the economists aiming to attain MUI could naturally attain the conservation objective. And Fishery shown that it's kind of true, but then there's difference in composition. Even the fishing intensity can be similar. And so each of these three reference points has its own practical application, and so it's essential to define the goal of the management plan. And I have shown that it is possible to use the mechanistic model to achieve that. And further research is needed to enhance the accuracy, but the potential is shown, and Fishery have the efficacy and simplicity to act as the intermediate solution to comprehensively assess the ecological consequence of fishing and even climate change in the future. Thank you very much.
Thank you very much. Perfect timing.
Thank you.
So, I will give the jury members the chance to ask questions. You want to go ahead, Miguel?
Sure.
All right.
I will start. Well, first of all, congratulations for the presentation. I really enjoyed it. I think it was quite clear. I understood all the concepts in there. Probably there were some slides that these slides appear just a few seconds and disappear. That's not the ideal. And also there were some slides with a lot of text with any figure that could actually summarize that text that could be shown to the public. You know? Figures are, in general, more recommended in this kind of presentation than just the text. You know? These are little things. I really enjoyed the presentation and it was quite clear. And coming to the scientific content of your talk, I think the model gives quite clear results, and you explained them well. But as all models, it has, I guess, it has some limitations. No? Could you please explain some of these limitations?
Yeah. So first of all, obviously, the species is not here. So if we want to imply it to, let me go back to this slide. If we want to imply it to really using it for ecosystem-based fishery management, we need to either make it more refined, or we just use it as a conceptual tool if we look at global target. But if we want to do it regionally or smaller scale, we need to adjust the equation and all the coefficient for that fishery. So that's the major limitation, and it can be quite sensitive. Like I haven't, like for me, I haven't conduct this sensitivity analysis because I don't have the time. Because we spend a lot of time just adjusting the model and stuff. But then, and the size class is one problem because I have 6 size class here for the bigger fish, 4 size class for the smaller fish. But then it's always a problem if you change these parameter, the result change as well. But which one is the better one? So it's difficult to find that stable point, which we attempt to do it here, like we did it here, but then if we change a little bit for a smaller scale, and it would be difficult to do that. Yeah, it needs a lot of time.
Let me point to one potential limitation that I don't know if you take into account. I don't know actually if it's a limitation, correct me if I'm wrong, but do you take into account the migration of whole stocks towards the poles?
There is no.
That's a big effect.
Yeah, I understand.
There are some fisheries that are moving to the poles.
Yeah, but then also it's super difficult for vertical migration. Horizontal migration. We do have a setup for vertical migration, but it's more complex. But also it depends on what's your aim of using these tools. Like this is never aimed to just look at one stock that will migrate from here to there. It's more like looking at a whole overall biomass distribution globally when under climate change. This model actually was used in FishMeat. I don't know if you heard of FishMeat, but this is part of FishMeat. So all other trophic, like there is some other marine ecosystem that use the empirical data, and then they give this biomass estimation, but we didn't know if that's true or not, even if it's real world data. So mechanistic model is also included in inter-region fishery comparison project. It's like a global project, and many institutes are involved, and each give their own biomass estimation. So in the end, we will have a curve, like a curve that from many models estimation, and then we can get an idea what is the actual change when climate change is there. Yeah.
I want to talk a little bit about the example that you put in your thesis about the Simian lake.
Yeah.
Well, as an example, it's a little bit weird because it's a lake. It's not the sea. Do you think that could have potential effects on the comparison you are doing? Because it's not the same system, right?
But then it's also a fishery. Like like in that paper, they compare really extensively with the neighbor. Like the lake is divided into half, and they compare it, they catch, and then I mean the fishing gear they use are they're just comparing because the fishing gear they use are different. So the size class they target are different. So it's mainly because it's a size-based model. So I don't think it's not relevant. I think it's because in that paper it is used. The size-based model is used to check like what is the difference in these two ecosystems. So I think it does represent this.
Yeah, I don't have more questions. Thank you very much.
All right, thank you. Fernando?
Thank you. Thank you very much, for this nice presentation. I really like it a lot. I realize that there is a lot of work here, and you know, I have some familiarity with this model. I work with Colleen.
Oh, Colleen.
And I know that you know it's not easy to work with this kind of tool. And the fact that you got all those results and you were able to assess all those hypotheses, for me has a lot of merit. So congratulations.
Thank you.
Having said that, I have two questions. Okay, one is a mere curiosity. That it's why did you change the forcing, the ESN that you are using? Or you are using a different ESN?
That's true. Okay. And this is because we are actually because the whole team is working together, and we choose to change this UFIC because UFIC is open access, so we can change the parameter in there. And so one of my colleagues, they are working on trying to give feedback to the Earth system model. So it's like so before, like now, I'm only using the output from the Earth system model. But then when he do that, I can actually, I don't know, all these fishing scenario, I can actually apply in there. So that's why we choose to use that one.
Okay, you know, there was like a follow-up. Perhaps if you stick to the previous protocol, then you can contribute at the end to the FISMI.
Yeah, but then I think this is not what FISMI want to see because I am varying the fishing mortality. For them, it's also difficult to compare between other models because what I'm doing is not what they're doing. Usually for MEM, what they do is they just put one single fishing mortality across the globe. And I don't think that's realistic. But then anyway, that's what they're doing. And this is different things. So that's why I didn't think about that. But I would rather see if my colleague can do that step, and then if I can use this information to go back to like have a full interactive model. I think that's more important to me.
Yeah, that's really interesting. And you know, my second question is a little bit more okay? You know, when I hear or see people talking about this kind of models, you know, what comes to my mind is like balance harvesting. You know what's that? Okay, no. For me, it's like something that I have seen from outside. Okay, but there is a comment that comes in and out, or that arises in those contexts that it's, you know, what are the species? Okay? And in the context of this particular, how did you handle the species concept with the economic aspects? Because, you know, when I go to a restaurant, I don't ask for a fish or, I don't know, in the size class between 2 grams and 20 grams with some olive oil and potatoes, you know what I mean? And I won't pay you or give the same value or pay the same for this kind of aspects. So I would like you to develop on those aspects. How did you handle it? What kind of aspects do you think that is going to improve?
I do believe that individual effort is less like the weighting of individual effort on this kind of issue. Like the more important part would be the whole management objective. Like when I was exploring this 3 reference point and I see all the case study in different regions, I just realized that like what the government choose or like what the international group they choose to employ is just it determines what you get from the fishery and determine what the fish you eat. But then the decision is they make it. Like so, in the end, it seems like we are making a choice between a better fish or less a worse fish. But then in the end, they choose it in the beginning. Like if they choose to, or they now want to have highest yield because we don't have food anymore. All the fish are fished out, and we choose to eat we need to, they change the management objective, and then we want more forage fish to be fished. We can't choose, then we have to eat more forage fish because we're not the fishery. Like, we're not the one who.
Way around, you know what I mean?
Yeah.
Because I want more expensive on a given kind of fish than the fishermen who are idiots. You have behavior.
Yeah.
They decide, I want to fish this because I will get a profit. Then the thing escalates a bit, you know what I mean? They start to talk with other people and say, okay, we have prices for that.
That I don't know, because that is exactly this discussion.
You don't know, nobody knows.
But this is
to talk about
this is the exact discussion here. Like, I demonstrate these, like max catcher, what was the composition in max catcher, and I demonstrate what's the composition of the max ones F, and I demonstrate that okay, economic yield seems like it's really close to conservation target, but how it employed and how it really actualized in the real world is so much more complicated. But I do think the two reference point that I said later, like the max range F, is actually a good indices. Like, we choose the fish and then we keep them low, like we keep the fishing mortality low. Yeah.
Thank you.
Yeah, just a short question. So, how this model performs when you compare this model with other models? Because when you construct the models, it's supposed to be some advantage or new knowledge.
So we made.
Have you made any?
No, like
comparison with
this
other
will be
models or
this will be the further research, because I explained before the fishing scenario that I apply here, other MEM is not doing them at all. So if I need to compare them, I have to apply this fishing scenario to those MEM, which I'll have to go through. Like, I have to understand that model and then apply this again and then compare the final yield output. So it's super complicated and it will take so long. I wanted to do that, but then I don't have the time. But then the starting point of this model, the biomass comparison, there's a slide about that. That the biomass distribution is agreeing with the previous paper, even though the Earth system model is different, and then we made some modification on the theta preference. So that one, and then we also compare the number, not just the map. So the starting point is the same, like it's similar to the previous paper, and then the previous paper, of course, is published, so it's extensively compared with other MEM, and then they're agreeing with each other.
Thank you.
All right, thank you very much. And we welcome our next presenter, Colbert Simmons. I want to remind, I'm pretty sure this was finished work on your and a submitted thesis on your case, but I want to remind also the next presenters, if your thesis is not submitted, please tell us before you start. But considering the work, I'm sure it is.
I'll do it.
You should find, Claude, you can close that window, and you should find your presentation here. Yeah. Just put it in full screen, and you're good to go whenever you're ready. Albert. Whenever you're ready, go.
The stream is working?
Yep. I'm still seeing the end of. Yep, you are there, you are live.
Wow. So, hello, everybody here in Oviedo, and everybody who joins from somewhere else using the new technologies. So ever since since Darwin, biologists have wondered at and studied the complexity and diversity which seems infinite, and individuals, organisms, differ widely, vastly in their ecology, in their life traits, in their evolutionary history, in what they eat, in their place in the food web, in their functional traits, in their environment, in how they interact with their environment, in how they interact with other species. It seems infinite and so complex. And here, the coming 10 minutes, we are going to ignore all that. And we are going to talk about generic organism X. So my name is Kobe Simons, and I welcome you to the wonderful world of neutral theory. So neutral theory has been developed by, well, not been developed before, but has been defined properly by Hubble. And it assumes ecological equivalence between individuals, regardless of the species. What does this mean? This means that they all have the same turnover rates, being that bird speciation and migration rate, regardless of the species. This, of course, means that neutral theory is mostly defined within a trophic guild, so individuals that compete for the same resource. But we will come back to that later. This also means that communities and community patterns are assumed not to be constructed by functional traits or niche occupancies or whatever biology. No, it's just random drift, random community drift, and that's it. These assumptions are philosophical assumptions. They are not scientific assumptions, so you cannot disprove them with the statistical tests, which many ecologists have failed to understand properly. This is also related to physics. This is actually a physics approach, where physics, and definitely the physics of complex systems, is not looking for a detailed description of reality. No. It's looking for a thorough understanding of the dynamics. And this is related to the spherical cow principle, which I'm sure many of you know. But we can discuss it in the discussion. So we are looking for universality. And what are the universal patterns? We have the species-area relationship, which tells us how many species live in a certain area. We have the species abundance distribution, which tells us how many species or how many realizations have n individuals. And we have the pair correlation function, which tells us how different discrete samples with the relative distance r from each other are correlated. And these are all spatial patterns defined on a spatial 2D grid, which is also, again, related to how physics works. So we can harness all those tools in physics to describe these spatial patterns. And now I'm going to talk about the models that I used. The first one is a mechanistic model. I'm afraid I will use different definition of mechanistic and so on. So on the spectrum that we just were shown, I'm all the way over here. And beyond. So we have a 2D spatial grid of a certain size, which is defined as the number of cells. And the cell size is arbitrary. You can do whatever you want. Within a cell, there are living individuals of different species, but we already know it doesn't really matter. Every time step, a species dies. A cell is selected randomly, and an individual is selected randomly within a cell, and it dies. Its place is taken by an immigrant from another individual. With probability m, this comes from the more neighborhood. With probability 1 minus m, it's taken from within the cell. And with probability nu, this immigrant becomes an entirely new species. So we have migration and speciation. Additionally, we have metabolic theory, which states that the rates depend on temperature according to the Boltzmann factor. So the turnover rates are adjusted with the Boltzmann factor. And we add that the number of individuals within a cell can vary according to a function of an environmental proxy which is capturing habitat complexity or something like that. Both the function and the environmental proxy are to be fitted. And it will depend on the taxa that you're looking at. Titensor and Worm used this to describe global biodiversity patterns. In the context of this thesis, the model is built from scratch in Python. Some developments have been done with regard to efficiency, but the main development is that it's used for smaller scales, regional continental scales. We start with the continuous plankton recorded data, which is data on plankton gathered over more than 60, 70 years, I think now, which is highly standardized by towing the plankton recorder behind ships. In this model, we only used 108 copypod species and presence-absence data only. The simulation is run only with temperature-dependent rates, because we have seen we use different type of scaling for the number of individuals within a cell with habitat complexity, and it is not a better fit, so we only use temperature. And these parameters for speciation rate and migration rate. The grid is arbitrary to capture a uniform distribution of samples. We are here, on the coast of Europe, part of Greenland, and United States is here. And the model captures actually quite well the main patterns. We have here the hotspots, here, and the lower diversity here. And diversity is simply standardized, so the maximum diversity is 1. A good theory works everywhere, so we also use it on North American breeding birds. I'm just going to discuss the data set. It's again almost 50 years of data collected in yearly census by layman experts, birding experts, across thousands of samples, transects in the United States. It has abundance data, so it's a really, really impressive data set. We can discuss it if you have questions, but I'm going to move on to the model performance. So actually, if we look at the residuals and the residual distribution and the scatter plot, we see it's well behaving. It's like no outliers, no systematic deviations, so that looks fine. However, if you look at this is a problem. Speciation rates of like 0.1, that's ridiculous. Titensor and Worm tried to have a biological hypothesis about that, but we tried to use a new prediction, namely the PCF. We looked at the PCF. Yeah, it's what you expect from other neutral models, that the correlation length scales with an inverse of the speciation rate. So species are in a cell and in the surrounding cells, and that's it. You, of course, also predict a completely ridiculous regional diversity of thousands of species, which is completely nonsense. If you look at the empirical PCF, which I will come back to later, yeah, so model is broken, doesn't work. So let's go to another model, which takes the correlation as the centerpiece. And this is now proper physics. So it's the binomial model. First, we take this form for the PCF, the pair correlation function, which is parameterized by rho and lambda, which both have dimension of length, and lambda can be considered the correlation length. This one is the modified Bessel function of the second kind of 0 order. It's just a function. And this one is the frequency abundance distribution, and is taken to be the negative binomial, which is parameterized by dimensionless r and xi, the Greek letter xi. both, so I throw them here, but both can be analytically derived from fundamental birth, death, migration, master equation defined on a 2D grid. It's a few pages of mathematics, so I'm not going to do it here. But it has fundamental properties. And this model is an innovation within the context of this thesis. And it combines the spatial correlation function of Azzaeli and others with the discrete sampling method of Tovo and others. We can use it like this, and I see that my time runs out, so I will just say that you can combine these equations, and if you have questions, we can go back to get these parameters on any scale, on any area with size r, which is just the square root of the surface area, given these parameters. This gives you the SAD, the species abundance distribution, and it gives you the species area relationship from fundamental principles. Additionally, we can also sample, and this gives you a disconnected area. The previous one was a connected area. This gives you a disconnected area because we have scattered samples, which is mostly the case in biological data. And again, we see that we have a negative binomial as a frequency abundance distribution. This follows from the mathematics. It's a prediction. It's not thrown. And again, we can calculate both the SAT and the SAR. This is the workflow. We can come back to that if you have specific questions. But comparison with data, in this case, the Barro Colorado Island data, which is an impressive data set of trees on a tropical rainforest on the Barro Colorado Island in the coast of Panama. And all trees are sampled, so we have information on the connected patterns. And we see that it's quite impressive. This is not a fit to the data, this is a theoretical prediction. So tell me if there are any other ecological theories that do this without fitting. This is the SAT in a Preston plot, which is logarithmic binning, and it's again quite good. Here there is still some work on showing it properly because it's the discrete problem with the discrete scale. On the North Atlantic breeding birds, it's not great, it's not terrible either. And you also only have to look at this because here it goes, there it's broken. Again, high correlation, this is something universal. We see high correlations in nature which can point to critical behavior. I will not go deeper into that, but we can discuss. The model predicts 650 species in the entire United States. And the estimates are between 700 and 1,000, so it's not terrible. Given the assumptions, so we require a homogeneous distribution of correlation, which is in the field that you have a uniform distribution of habitat. So yeah, so more or less the same habitat, no clear distinctions, yeah. And we, of course, have the ecological equivalence, which means competing for the same resource. This is one of the birds in the data set. This is another. So it's actually impressive that it works at all. So I mainly discussed these. The mechanistic model is a reasonable correlation with alpha, local diversity. It's a completely unrealistic nonsense PCF, so beta and gamma diversity, and it has some impractical degrees of freedom which are not nice from a theoretical point of view. The binomial model, actually very accurate predictions, regardless of the completely ridiculous assumptions. It's an analytical and progressive formulation, which means you can calculate and do quantitative predictions without fitting, and even if it fails informatively. Grammar. And then it's impractical for incomplete data sets. We tried it on the plankton data, which only presents absence, and it doesn't work. So there is still some work to be done. Thank you very much to these wonderful people who have shown me science and real life. This is an index of my images and the main references. And I now surrender to a room full of ecologists and biologists who are frustrated because they had to listen to 10 minutes of neutral theory.
Thank you, Kuba, for nearly perfect timing.
No.
You went a little bit over, but the thing is you managed to adjust. Like as you saw, you were taking a bit too long, and then you skipped something, so that's good the way to handle that. Thank you for I found that super interesting. It's unusual thesis and unusual talk for MBRC.
It has been approved, and I followed my.
I'm not criticizing it. I'm just I'm just mentioning it. I think it's super interesting to have this kind of work. I'm gonna let Nico start.
Sure, thank you very much. Yeah, very nice presentation. I really like how you address the public, how you presented your results. You simplify a very difficult topic, actually. But I'm not. I don't feel any confrontation with you or anything like that about the way you have presented your results. I don't see this fight against physics and biology here.
There is no fight because I use physics to explain biology.
Exactly. So I think.
But the theory and the models are completely against everything that mainstream biology assumes right now. Every article that uses neutral theory has to start with like 2-3 sections of motivation why we use this. And that's ridiculous. I don't see any niche theorists defending niche theory, while it's actually a lot less defined than neutral theory.
Yeah, that's probably true, but well, you have to understand that we have come a long way with theories, niche theory, for example.
But I think that's also a difference when a physicist talks about theory. It's not the same than when a biologist talks about theory.
Well,
I can give you some quotes on that.
We can discuss a lot about that. But some of the complexities that biology want to address and to present to the public, those matter in models.
Of course.
For example, when you explain the migration rates to a new place that is left by a species that is gone, that's affected by lots of variables, not easy to simplify at all. It's not the same that this process happens after a massive extinction or just during a normal time.
And the model is an equilibrium model, so it
will capture that.
No. It's an equilibrium model, so extinctions and so on. It assumes that nature is an equilibrium, which it is not. The question is whether that matters.
So yeah, actually I think it's a different work, the one that you have presented here, challenging work. I would like to read it actually. I don't have the opportunity, but I would like to read it because this deserves reading to properly understand all what you did. No? And yeah, I'm looking forward to read it actually. And yeah, I don't have.
Yeah, I enjoyed actually quite a lot breaking the models. So.
Yeah,
that's also something. That's something that the physicist does. He gets a model from somewhere else and then he tries it on, or he or she tries it on something new and then it fails.
Yeah, that's all I have to say. Thank you very much.
Thank you.
Thank you for the presentation. So, if I understood your neutral model describes one, the observed distribution of an abundance of the species, right? And still, my question is, And your conclusion is, which are the factors responsible for the global distribution patterns?
Yeah, so then we go to the mechanistic model and the work of Tietensor and Wern, and they conclude that temperature is the main factor. And if you look at the distribution of habitat complexity and how many individuals can you get in a certain cell, that's actually quite a similar pattern as compared to temperature. So it also increases towards the equator. So they focus on the latitudinal gradient in diversity, and they can explain actually for quite a lot of taxa with correlations of 90% and more the diversity patterns. But so it's a good descriptive model for diversity. But if you then look to a new pattern, the PCF, it's clearly nonsense. So it's a descriptive model, it's not an explanatory model. So, and on the fundamental theory level, we are interested in explanatory models. So then that's why I moved on. So after 2-3 months of work on the code.
And could include the size of the area of in your models, and that's at different latitudes, different environments, different temperatures, but it's also correlated with the available space. So
yeah. That's captured by the scaling of the number of individuals, according to Tietensor. But we do not like that, because you get what you put in. And that's from a theory point of view, it's just fitting. It's not. It can be non-trivial. So, for example, the copy pods are clearly driven more by temperature, and the birds are not, which is something you expect from an ecological point of view. But it's still just getting what you put in.
Fernando?
Well, thank you very much, Kobe. I don't know, I remember you from the classes, and I really like your presentation. You know how to engage the public, how to keep all people attention. I had some questions, and the twist that you introduced in the middle of the presentation, you just remarked all the things I was going to say. So I will just ask some very specific details. Okay? At the end, your model is a binomial or a negative binomial model?
I called it the binomial model because the frequency abundance distribution is a negative binomial. Okay. But then we introduce sampling, which is captured by, so the probability of having an individual of the total individuals in the sample is binomial if it's random. So that's why I called it a binomial model, but yeah, it's just a name. I first used the name of the authors, and they said, don't do that.
That's weird.
They both did, so yeah.
I will keep negative binomial. No, that will ring in the minds of many people.
Yeah, of course.
Parasitoids with,
because it's also a good fit to the data.
Yeah, even in fisheries, it's a model that now and then it comes up. Okay, it has some characteristic that it's good for modeling situations where you have many seals.
But it's discrete, so it's not very nice in the calculations. The gamma distribution is more convenient for calculations, which is the continuous variant of the negative binomial.
So, just to try to understand a little bit more about what you do. So, the predictions of the model are just large-scale emergent properties like the SID.
Yeah.
The SR.
So the SAT is just the SAT on different scales. That's just the scaling, a spatial SAT, and the SAR is a spatial.
Okay. Rho is the size of the domain.
Rho?
Rho.
The parameter rho of the PCF.
Yeah.
No, it captures how large is the correlation on any scale.
Okay. Lambda. No.
Lambda, it can be considered a correlation length.
So that would be somehow related to migration rates.
Yeah, probably. Yeah, it's in the model. Lambda can all be reduced to the fundamental birth, death, and migration rate.
Okay, I have everything I need for my last question. Okay, so I was freaking out and that you were talking about the speciation. I know that it's from this one. Okay, don't worry, I'm not saying anything extra. But you know, in terms of dispersal, you know, when you see that, what I'm thinking about is, you know, the roof stream coming out, all the North Atlantic Drift, a huge front, subpolar gyres, tropical gyres, you know, it's like a divide. Okay?
Yeah.
So, you know, two things, okay? To what extent do you think that your model is correct, or that the patterns that you are analyzing actually emerge from speciation, migration, and this kind of, you know, like
Random drift,
scale
random drift.
processes, and whether, you know, the particularity of, since we are in a master about marine science, the particularities of the North Atlantic and oceanic currents and all that kind of stuff, when you talk about plankton, maybe the explanation for the lack of fit on the ocean.
Well, the fit is actually quite good.
The fit is good.
Yeah, you expect, this is
No,
this is quite good in the context of these models and the context of
incomplete data. That with incomplete
yeah,
data,
but I do not agree with what they said, that it's a good that the model is capturing what happens. No, actually, what you expect as a physicist, if you do it on a global scale and then you try it on a continental scale, regional, local, it should fail somewhere in between. If it doesn't, there is something wrong. So the fact that it actually gets the same correlations as on a global scale is not good, because you expect different processes on different scales. So that's why we started to doubt this model. So I completely agree with you.
But what about, you know, for instance, for a tiny planktonic species, do you think that, you know, the kind of pattern that you're observing,
so the above,
speciation and
so the one on top, yeah, on these scales there will definitely be other processes, definitely. But on a global scale, I'm quite convinced in saying that doesn't matter.
Thank you very much.
Thank you very much again. Thank you for the presentation. I'm going to repeat, but you managed, I think, to engage in an efficient way a difficult subject matter for most of the people in this room in a good way. And I'd also be interested if you if this gets published, or if you want to share that with us, I'd be interested in having a look at your work with a cool ad and time to think about it. Thank you again, Kobe.
Thank you.
Whenever you are ready.
Okay, good morning everyone. My name is Abril Reynés, and I'm going to present my master thesis, which is called Long-Term Impact of Turbine Presence on Macrobenthos Communities in the Belgian part of the North Sea, which has been supervised by Professor Dr. Ulrich Breckmann and Professor Dr. Jan van Aberbeke from Ghent University. Offshore wind energy is a type of renewable energy that benefits from the high wind current speeds that we find outside of land in the sea. And as you can see in this graph, it has been increasing throughout the past decade, and it's expected to increase more in the future. The European countries that are ranking this offshore wind energy development are Germany, Belgium, Denmark, Netherlands, and Sweden. In this map, you can see a little bit where these offshore wind farms are placed in Europe, and as you can see, the North Sea has a big presence of turbines, with almost 6,000 wind turbines already established in European waters, 126 offshore wind farms, and with 13 countries that already have this energy development. Most important, in the future, there's an expected increase of offshore wind farm development. As you can see in this map, the areas that are colored with blue colors and purple colors are future areas suitable for development, or future areas where offshore wind farm is planned. So with this future increase, it's essential to understand which impacts these structures have in the marine environment, the marine ecosystem, and to be able to monitor these impacts. In Belgium, offshore wind farm corresponds approximately 8% of the total electricity that is generated. And as you can see here, in the Belgian part of the North Sea, there's one existing zone that was created in 2004, where there are 8 wind farm projects already established. And 3 years ago, there was a new zone that was created for future development. As you can see, is this new zone. So what happens when there's an offshore wind farm that is placed in the marine environment? It's a complex process and a lot of things play a role, but there's an artificial structure that is placed in the sea, in the marine ecosystem. This artificial structure modifies the hydrodynamic regime, which modifies the granulometry, the seabed topography, and the seabed permeability. On the other hand, the areas where the offshore wind farms are placed, the vessels cannot enter those areas, so there are areas which trawling is banned, so there's no fishing. And on top of that, these artificial structures are colonized by organisms, which these organisms produce fecal pellets that are high in organic matter, and these fecal pellets are transported into the sediment, into the sea bottom. So all these factors together, what they do is they change the biodiversity of the area, and they also modify macrobenthic communities. So here is a little bit what I explained, but with the diagram. As you can see, we have the turbine that is colonized by different animals, and then the water current also changes. So one way to monitor the impacts of offshore wind farm is to study macrobenthos. Macrobenthos are organisms that live in the seabed, and they are bigger than 1 millimeter in size. And they are important because they play a key role in transferring energy to higher levels of the food web, for example, affecting commercially exploitable fish. They are also highly dependent on the sedimentological characteristics, which are characteristics that the turbines might change. So what we realized in this master thesis was that the current research that was done was based on a short-term studies and on a small scale. So there was a need to find robust patterns over space and time, and more in a long term because of this future development. And we also realized that if we want to do long-term study, natural fluctuations of macrobenthic communities and the situation of global warming has to be taken into account. For that reason, the objective of this master thesis was to do a long-term study of 13 years in the macrobenthic communities in 2 offshore wind farms, and to investigate how macrobenthic communities responded to different predictor variables, and some predictor variables were related to the presence of the turbines and others to the climate. So there were 2 main hypotheses driving this master thesis. One was that we expected an increase of abundance, richness, and diversity of macrobenthic communities in the vicinity of the turbines because of the changing conditions. And we also expected a shift of macrobenthic community towards a more diverse community, which is called the Abra-Alba community, present in the Belgian part of the North Sea. And we also expected that natural fluctuations like sea surface temperature and also climate index could be able to explain long-term variability that was not able to explained by turbine presence. So this is our study area. As you can see, this is the Belgian part of the North Sea, which is this small part of sea that you see here. And in the Belgian part of the North Sea, there's an offshore wind farm concession area, and we studied 2 offshore wind farms, Sea Power and Belwind. They are 2 offshore wind farms that are located at different distance from the coast. They were built at different years since construction. They have different foundations. As you can see, Belwind has monopiles, and Sea Power has jacket foundations and also 6 gravity-based foundations. And they are located at different sandbanks. They are surrounded by different currents and water masses, so they have different environmental conditions. What we had macrobenthic data identified up to a species level that had been collected via scientific divers and via van Bingraps through a research vessel. And also we had abiotic data that contained organic matter, grains, and grain size. All this data had been collected from 2008 until 2020. And what I did was to compile and standardize all this data and add some additional predictors to the data that we already had, that it was water depth, years since the offshore wind farm was constructed, distance to the nearest turbine, sea surface temperature, and 2 climate index. which were the North Atlantic Oscillation Index and the Atlantic Multi-Decade Oscillation Index. Once this data was all compiled and all these predictors were together, we went through a data cleaning process, and at the end we had 564 samples, which looked a little bit like that. In this graph you can see in the y-axis the sum of the samples and the x-axis different variables. Most of the samples were taken in autumn, and via Van Veen graphs you can see at different depths and distance from the turbines, because all the data came from different monitoring programs and throughout different years, as you can see here. So then, when we had all this macrobentic data, we needed to find patterns. We needed to find what was happening to our macrobentic communities. So for that reason, we did a statistical analysis. We did univariate analysis via general additive models, GAMS, and we calculated 3 biodiversity index, species richness, total abundance, and Shannon diversity. So we did 3 models. How the GAM works is that we had our explanatory variables, which were the predictors that I added and the ones that we had. And we wanted to see if these explanatory variables had a significant effect on our response variable, on species richness, for example. So GAM models all these, and then at the end you end up with a model, which is a species richness, for example, depends on these few variables. And then we did a multivariate analysis. We used a merging technique that is called many GLMs. And what this technique does is that it fits a general linear model for each species on your data set. And then what we did, we used the ANOVA function to find the significant species for each explanatory variable. And then to visualize this, we used coefficient estimate plots. Now I'm going to present my results and discussion. I merged univariate and multivariate analysis for a better understanding of the big picture, and I divided it in 4 main predictors, which were fine sediment fraction, total organic matter, local factors, and climate effects. So fine sediment fraction appeared to be one of the principal structuring factors of macrobentic communities, meaning that it was one of the main predictors, and it was always correlated with higher species richness, abundance, and diversity. As you can see in this graph, in the x-axis we have the fine sediment fraction, and in the y-axis we have the smoother for the species richness. So here we can see that as species richness, as fine sediment fraction increases, also does species richness. And it was the same with the other biodiversity index. We also found that in the multivariate analysis that were 51 species that were significant for fine sediment fractions, and that their abundance increased with finer sediments also. So what we were able to see is that there was a transition from the original macrobentic community that we found in the area, that it's not a poor community, which is called the Nephthys cirrhosa community, and that was evolving to a more richer community, which is the Abra alba community, that is a macrobentic community characterized by more finer sediments. So why is there a change in the sediment in the turbine? As I said before, it's because when we place a structure in the water column, then the water column cannot flow as it flew before, so we are creating areas which are more sheltered and areas which are more dynamic, and then there's the position of fine sediment in the vicinity of the turbine. On the other hand, we also found that total organic matter was also correlated always with higher species richness, abundance, and diversity. As you can see here, there was a linear relationship between total organic matter and total abundance. So as total organic matter increases, also total abundance of individuals did. However, it was not as important as fine sediment fraction. The multivariate analysis just found 3 significant species that were responding to higher levels of organic matter significantly. And one of those species, though, was Laniche conchilega, which is a species that is characterized from the Abra alba macrobentic community, that is an ecosystem engineer, and that it benefited indeed from high organic matter. Why is there an organic matter enrichment, and why maybe it's not as important as fine sediment fraction? The organic matter enrichment is due to the fauna, the falling fauna that colonizes the turbine, that generates these fecal pellets that are exported in the sediment. And because this sediment gets finer, it's less permeable, so the organic matter gets more easily trapped. However, one explanation of why organic matter is not as an important structuring factor could be that there's not a clear as a clear pattern as fine sediment fraction, because some studies in the area that were done in the same offshore wind farms a few years ago could not prove that there was an organic matter enrichment in Bellwood, for example. Then we found very interesting results that were about local factors. So we saw that some biodiversity index were not acting the same in different offshore wind farms. So for example, species richness in sea power increased towards the distance of the turbine, as you can see here. And it didn't have an effect in Bellwind. So species richness basically stayed constant. And in terms of years since construction, total abundance increased with years since construction in sea power, but it was completely opposite in Bellwind. It decreased with years since construction. These results could be due to the different foundations that we find, jackets and gravity-based foundations in sea power and in Bellwind monopiles, which could affect the fauna that colonizes the turbine and also the hydrodynamics, and could also be due to the fact that there are different currents, different systems, different energetic systems. And last but not least, the climate effects, which was the more new part of the study. We saw that in all the models, there was always one climate predictor included. For the species richness, it was the AMO index, a climate index, and for Shannon diversity and total abundance, it was sea surface temperature. But as you can see, even they were important predictors because they were included in the model, the trends are not clear. With the species richness, it seems like species richness oscillates with AMO. And for total abundance and Shannon diversity, as you can see, the patterns are not clear. This could be due that 13 years is too short to be able to see natural variability and to be able to see effects of climate, and also could be due to the fact that most samples were taken in autumn, so we couldn't have a whole gradient of temperatures, and also we missed seasonality and the complete temperatures. So to conclude, as you can see, offshore wind farms are a complex matrix and a lot of things are happening at the same time, but however, we've seen that we haven't found a stable state after 13 years of turbine presence. So macrobenthic communities are still changing, maybe because of the presence of the turbines or because of natural fluctuations. However, we found that the turbine-related effects, which are grain size and organic matter, are the main predictors of macrobenthic communities, and they are changing because of the presence of the turbines. No clear patterns related to climate effects emerge. However, we saw that it's important to take into account these predictor variables, these predictors from climate change, and when doing long-term studies, they have to be considered. And finally, we realized that the impacts might depend on local conditions, so it's important to do future research in different locations and regarding different foundations to see which are the things that are changing, with the whole aim to do a cost-effective management and to improve our knowledge of these structures in the marine environment. This is my reference list, and I would like to thank my supervisors, my friends and colleagues that are here, and everyone that has made this possible. Thank you so much.
Thank you, everybody, for this dynamic presentation made within a second of the allotted time, I must say. So, perfect in that regard. Julio, would you like to start?
Yeah, sure. Thank you very much for your presentation. And could you please put the previous slide? Yeah, no, that one. So you say that no clear pattern related to climate effects. That's but you have not evaluated control areas. What happens with controls? Yeah. What happens with areas without turbines? Mm-hmm. What is going on there?
Yeah, so a lot of the data that we had also had a control area which was another sandbank that is similar to a sandbank. However, because we wanted to test turbine presence, so the effects of turbines, we just used the data that was within 1,000 meters from the turbines, because this is what is, according to literature, this is what is understood as the area of effects of the turbines. So with the purpose and context of this study, we decided to avoid points and sampling points where that way were very far away from turbines and also control areas. However, it's true that depending on the research question, I would say it's maybe necessary to account for the control areas, but because we wanted in this thesis to just see how macrobentic communities evolved with the context of the turbine presence, we decided to not take into account those points.
That's my point. If you're trying to evaluate the effect of turbines in benthic communities, you need to have some control area without turbines, not 1,000, 1 kilometer away, several tens or 100 kilometers away. So I think that you can evaluate if patterns are related to climate or if you have control. So with only evaluating the turbines and the environments, I think that that's can be wrong, right? So, and usually people say that the positive, or turbines have a positive effect. Do you agree with that?
I think the impact of the turbines is very complex. It's true that there are some positive effects. For example, trolling is prohibited. And for example, the Belgian part of the North Sea is an area which can be fished everywhere. So in fact, the areas where the offshore wind farms are placed are areas free of fishing that we don't find in the whole part of the sea. So this can be a positive effect. But also sometimes more doesn't mean better, no? So that species richness and diversity is increasing. We still need future research on that to be able to evaluate how the food web and how the ecosystem changes when the macrobenthik community is evolving towards a more richer one. So if you ask my opinion, I would say that it's difficult to say if it has a positive or a negative effect, but what I think is that we need to move towards a more green future and we need to move towards renewable energy. So the best thing is to try to understand as maximum as possible how these structures are going to affect the sea and try to mitigate or try to install them in the best way.
Yeah, I agree with you with the trolling ban is the positive part, but modifying an ecosystem, I cannot say that's a positive effect.
Yeah.
So you're assuming that the previous ecosystem was wrong, so we are important, so we are improving the ecosystem with the turbines because we are increasing the number of species, we are increasing diversity, but sometimes low diversity areas are part of biodiversity, global biodiversity. So you need to be very careful with evaluating or saying that that's positive, that's negative. So far the only positive effect I see there is trolling.
The trolling, yeah.
The ban.
Yeah, I
So
agree.
Thank you.
Right, thank you. Fernando?
Okay. Well, thank you very much. It was a nice presentation. It was very clear, the objectives of the results, the method. I really appreciate it. I don't know more about PACI Napoleon, okay, but I would strongly recommend you to incorporate those extra data. If you have a control area, incorporate it. Because I think that caught my attention a lot was that you were exposing the conclusions and the mechanisms for the good results that you with or the sampling results that you find very strong. Certain them with a lot of confidence that, for instance, this kind of basement for the turbine is better than this kind of stuff. And I'm not completely sure. Perhaps in one place something is going on, and in the other location another trend is occurring for whatever reason. That in those locations, having a control area is very useful because if you find the same trend in the control location, then something else going on. If you have something different, then you have testimonial evidence. But anyway, moving a bit forward, do you think that I heard some stories, okay? I think that you can ban bottom drawing, and perhaps you should, okay? And you don't need the excuse of putting there a turbine map. Do you think that there can be any effect of the turbine fluids as an attractor to fishermen, outside or in the surroundings of those areas, or that it can be the other way, fishing and then there is more fish around, using the fields as a refuge?
Yeah, so it's again complex because you are banning trolling from an area, but then studies made in the Belgian part of the North Sea have seen how like fisheries have been redistributed, and they like to fish more. So there's more fishing efforts in the edges of the offshore wind farms. So yeah, I think it's good that there are areas which fishing is not allowed, because then the diversity and the macrobenthic community can recover. And also the turbines sometimes act as nursery fish areas, but however, it's nothing is good or bad, it's again complex because fishing effort can increase just in the edges of the concession areas.
Well, something very specific, okay. If I understood it correctly, you got the same results from species richness. I don't remember your second.
Total abundance?
Total abundance and salmon diversity. There are some kind of rumors in the literature that you should not use the salmon index anymore, okay, because it's sensitive to sample size or something like that. It's also very good. Like instead, perhaps it is better to use hill indices. Hill numbers, sorry.
I didn't know this.
But at the end, why do you think that you get the same response, if I got it correct?
So the models were quite complex, and sometimes the response variables varied in some things. But I do think that we get the same response for these big predictors like grain size and organic matter, because I really think they are drivers of these macrobenthic communities. So, for example, they are a little bit correlated, no? So if species richness increases, there must be that this macrobenthic community likes, so it's more possible that total abundance has increased and also channel. So there's a more diverse community. So this would be my guess of why I think some predictors have the same response to the variables.
Thank you. Maybe just to follow up on that, the thing like Shannon varies on a logarithmic scale, and if you all use yield numbers, you put it back on a linear scale with understandable units for us biologists, which are number of species. So it might be easier. It's easier to understand, to grasp, and sometimes in analysis it's better to use simply the exponential, basically, of Shannon or of Simpson diversity. Okay.
Well, first of all, congratulations for a very nice presentation that you did. I really enjoyed it. You engage with the public here. You were able to explain the main results of your thesis here. I really like it. And about the scientific contents themselves, I think they are very interesting. I have some little doubts, for example, regarding the stats. You put there a lot of different variables in your GANs. Did you check that you didn't have any collinearity effect in between the different variables? So for example, if I understood well, you have taken into account SST, the surface temperature, but you also took into account the climate indexes, no? The NAO. So, did you check that?
Yeah, we checked for collinearity, and we found that, for example, median grain size and fine sediment fraction were correlated, and so we excluded median grain size, and then we checked for collinearity, and so we did the matrix and also the variance inflector factors, and everything was
okay. Well, that is good. But specifically, did you find any correlation between SST and the climate index? Do you remember?
No, the climate index were not correlated, but
SST.
Yes, so I had the average sea surface temperature, and then we calculated also maximums and minimums to account for extreme events, and such surface temperature was correlated with maximums and minimums. So we excluded sea surface temperature and we stayed with minimums and maximums.
Okay, now I get it. Okay, cool. And regarding other aspects of your thesis, what they told you, and I think it's good that they have told you that to assign the term negative or positive to an effect, we shouldn't do that, no? Especially, we cannot consider that an increase in diversity is good for itself, no? Maybe you are in a low diversity community, and when you increase diversity, it's a change that it's not natural, and that should be enough to avoid it, actually, to try, or at least to try to avoid it in some way. But of course, as you say, we need the green energy, right? Do you think with the actual wind farms that you have there in Belgium, do you think it's, you mentioned that it's 8%, no? Of the electricity that is provided to Belgium by these wind farms. Do you know anything about the expectations in the future, if that is going to increase to 50%, for example, or something like that? Or?
The new area that has been established is to reach the European target that says 17.5%.
17?
Yeah, so almost 18% of the total energy of Belgium provided by offshore wind farm.
And do you know anything about the actual trends in wind velocity in that part of the coast?
I know that the European targets are very high for the 2030, 2050. So if they want to reach it, offshore wind farm development has to increase.
Yeah, but maybe nature has other plans, because I think actually wind trends forecasted for that part of Europe tells that
wind speeds are going to decrease.
What?
Sorry?
The last one, please.
The
ultimate
question,
please.
Okay. Yeah, I'm not going to. Just a brief question about if you expect any spillover effect of the nutrient and organic matter effects that you see in the wind farms. If you expect that to spread out a little bit, 1 kilometer around or 2 kilometers around the area, because it's said that for fishes, it's not demonstrated, but it's said that for fish, they expect accumulation of fish in the wind farm and then a little bit of a spillover effect around it. Do you think that for grain size and organic matter that is going to happen or not?
I would say that this can in fact happen. I don't know if how much or if a kilometer, but what we have been already seen is that fine sediment fractions are increasing also in a little bit farther away from the turbines, maybe 500 meters or something.
Okay, that's all.
Thank you very much.
Oh, and you have some cheers from the chat, from Ulrike, Jan, Alphonse, and Irene. Just so that you know.
It's okay if I have a timer here as well?
Sorry?
It's okay if I have a timer here as well?
Absolutely,
Okay,
just to show you.
Yeah, okay, thanks.
Absolutely. Whenever you are ready.
Okay.
Elia, the floor is yours.
Okay. So hello, my name is Elia, and I am welcoming you to my thesis presentation. I am excited to share with you the project that was supervised by Dr. Giuseppa Buscaino and Dr. Maria Serraro at the CNR. There, I conducted an analysis of soundscape components and their seasonal patterns in the Kongsfjorden at the Svalbard archipelago. So let's break down the first part of that title and explain what a soundscape is. So a soundscape is an ensemble of sounds in a given environment. This means it includes biotic sounds such as cetacean calls, abiotic sounds such as wind and rain and even the melting of ice, and anthropogenic sounds such as shipping. Now, depending on where we are, if we're in the tropics or maybe up in the Arctic, the different components that are emitting sound are going to be more or less present. And because Soundscape gives us all of the sounds, it allows us to get a holistic picture and gain a better understanding of all the components that an ecosystem relies on to function. So we can look at the ecology of populations, community structure, species abundance and distribution, and presence of anthropogenic factors, for example. And all these fall under the term of ecoacoustics. And I find this image to be very representative of what a soundscape is, and something that we can see and understand visually. And it does match up a little bit with my study site, and so some of the sounds we could hear are, so I would put, for example, this hydrophone in the water, it would do passive acoustic monitoring, and we could hear, for example, the rain, as I mentioned, ice-related noises, the shipping, wind. We, of course, have plenty of cetaceans and fish, to name a few examples. And so understanding the frequency range that each of these components has can help us translate this audible information into something a little bit more visual. And especially having large-scale and long-term monitoring will allow us to see the effects that climate change or rising temperatures in general may have on an area and its marine life. And so to move on and explain a little bit more in detail about my study site. So we go above Norway to the Svalbard archipelago. And here I have my Kongsfjorden, the fjord I analyzed. And there is a glacier at the inner part. And it does influence the fjord by the freshwater runoff from the melting of the ice. We also have Atlantic flowing in. And these two are going to mix, creating interesting hydrodynamics, and they make for a unique environment that attracts plenty of different species. And so up there is the placement of the static autonomous hydrophone, which was recording for 2 minutes every hour. And so the aim of my study is to characterize the Kongsfjorden soundscape over the course of 1 year, so from May 2014 to April 2015, and estimate annual ambient sound levels and assess seasonal variation. I also wanted to emphasize the importance of rapid assessment because I had a large amount of data to treat in a small amount of time. And so by using straightforward methods, we can gain a comprehensive understanding of sound patterns, which can help contribute to baseline data and aid in future comparisons and accelerate the decision-making processes, which is important for implementing conservation strategies, which is especially valuable in the Arctic, where the effects of climate change are occurring at a more rapid pace. So how did I do all that? This figure here shows a little bit of the methodology that I used. And so I have the recordings that I received from my research center, and from there I did 2 different kinds of measurements. Well, the first was energy measurements, which is basically like the physics of sound, and this, as I mentioned before, is looking a lot into frequency. And so the first one is the broadband analysis, and this looks at all of the frequency range together and the sound levels of that. Then I do a 3rd octave level band analysis, and this splits the frequencies into specific bands, and then I look at the sound levels of those. And this was conducted using an algorithm called PAMGuide in the software MATLAB. The second aspect is an acoustic index, so a numerical value that gives us information on specific sound characteristics. I chose the acoustic complexity index, which looks at how modulated a sound is, which helps us highlight biological signals, which tend to be more modulated than non-biological signals. And this was conducted in R. I also took external data, so everything here, like species vocalization count, which I obtained from previous studies from the research center. I downloaded environmental data from NOAA and Copernicus, and then I also had vessel presence. Now, vessel presence I actually obtained from my recordings, and I ran it through a vessel detecting algorithm in MATLAB. Now, what I did is I took all of these variables and I tried to analyze it against these 2 analyses by noticing if we could see these variables in the sound patterns. And I also wanted to see if that would be noticeable on a seasonal scale. Then all 3 of those can and will interact together. And so I look at how that interaction contributes to the variance of sound, but only for the energy measurements. And so this was done using Kruskal-Wallis test, correlations, and in the end, general additive models. So to go a little bit deeper, so for my biological signals, again, the species vocalization counts I had were from fin whales, fish, seals, and walruses. The shipping part I obtained from this vessel detecting algorithm, and my geophonic components I had were wind, rain, and sea surface temperature, which I used to describe ice-related noise. Now, as I mentioned before, each of these variables has a unique range. And so I had to choose a toll band to represent that variable, and so the toll band is a range, but we usually call it by the central frequency, and I tried to pick one that would be most representative for each variable. I do want to point out that for the shipping, I picked the 63 Hertz and 125 Hertz, which is recommended by the European Marine Framework Strategy Directives to investigate shipping. I did the same for the ACI band, but I had a little bit more liberty in the range, and I tried to encompass as much of the range of each of these species as possible because, again, ACI was only used on the biological component. So what I did is I took each variable, correlated it against the toll bands, and then I correlated it against the ACI bands. And then I looked how these two varied seasonally and how they varied seasonally in the presence and absence of each variable. All of this analysis gives me, we're going to start easy a bit first, just the annual trends and seasonal variation. But what does this fjord sound like, and what are the sound levels? I just put here the broadband analysis, so all the frequency together, because each toll band showed the exact same pattern, and so I find it a little bit easier to look at. We have a rolling average here in the dark line, and I want to point out this increase after October in sounds. And this is reflected in the seasonal variation, where we do have this increase in autumn and a bit of these lower levels in summer. So, moving on to our first component, the biological component. This, again, is just the vocalization data that I got, and we can see that it is occurring at very specific moments of the year and for a certain amount of time. And so the rest of the year, there's all this variance that is kind of hard to account for sometimes. And so while looking if the toll bands could highlight the species presence, no significance was found, probably due to this low occurrence of vocalizations at certain moments. However, the ACI did show promising results for fin whales and for fish. So in the blue boxes, I show the values when vocalizations are present versus absent. And the months that are significant correspond with the months that most of the vocalizations can be found. And so again, the higher the ACI value, this will be indicating biological signals. For the anthropogenic component, so I use this vessel detecting algorithm, and the output I get is this, where it shows this number of peaks. And so anything above 0 can be counted as vessel presence. This algorithm on my recordings showed a sensitivity of 92.8% and an accuracy of 73%. And so the positive correlation that I could find was looking at the positive peaks and the 63 Hertz and 125 Hertz toll bands. It is not that strong of a correlation either. So this vessel detecting algorithm was inspired by Rice and al. 2019, who created it around recordings that were not taken in the Arctic. And so I did manually check these, and that's where I got these values. But I found a couple false positives, whereas their algorithm didn't have any. And so I think that this probably is going to have to be reviewed and improved to be able to better apply it to Arctic recordings. Our final component, the geophonic component here, shows some nice graphs, nice correlation graphs between the wind speed and the sound levels, and as well with the temperature and the sound levels. In orange, we have the 1 kilohertz toll, which is what I use to describe the environmental variables, and in blue, we have the broadband, so all of the frequency together. What we can see is that there is a correlation between the increase in wind speed and increase in noise. It actually is about 10 decibels increase, which can be quite considerable. However, we do have a negative correlation for the temperature, so showing a decrease in noise with increase in temperature. And this, if you guys remember, in the seasonal variation graph, we did see those lower levels of sound in summer. And what this does and what this tells us is it highlights this, the high seasonal influence that occurs in this area. Now, up until now, we've always been investigating this component by component, but this doesn't really reflect reality, as there's going to be many other variables that come into play and that they might interact with each other as well. And so here is where I conducted my general additive models. So I had a model for each of my toll bands. What I did is I added the variables that could potentially influence at this frequency, and then after messing around with it, I selected the one that had the lowest AIC and the highest deviance explained. And the first thing to point out is how hour and month is very present in each of my models, showing again the dial cycles and this seasonality that is very important. While the toll bands were not able to highlight the biological species, they do still participate in contributing to the variation of sound, especially with the model with the highest deviance explained being 800 hertz toll, with only 3 variables, 2 of which were bearded seal and walruses. However, kind of going back to the slide before, this importance of all the abiotic factors is clearly seen as well, where the sea surface temperature and the wind do come up very often in each of these models. So, after all of this, to sum everything up, after looking and characterizing the annual ambient sound levels and looking at the seasonal variation, which again can be used for future comparison, as my year was done in 2014, I was already able to look at another study done in the same year. And they also found this increase in October. But if you look at the year afterwards, 2015, they show the opposite. Louder levels in summer and quieter levels in October. If we do think about it, it does make sense. In summer, we will have the noise from the melting of ice and this increase in shipping. And so while there's different, we can also have like anomalous ice melting occurring at different times of the year. It is also important to continue this long-term monitoring so we can account for this natural variation and notice the changes. And so while the toll bands didn't highlight the biological factors, the ACI was able to highlight the presence of fin whale and fish vocalizations. So overall, the strong dominating factor here is the abiotic factors. And this is going to be very important in the coming years with the increase of temperatures, which, or I think as we mentioned earlier, we might have changes in distribution of species. And if we have rising temperatures, we're going to have more ice melt, which means that the presence of anthropogenic activities can occur more days of the year. And we all know that this noise is detrimental to marine life. And so by monitoring the noise levels and the climatic changes that this ecosystem is facing and its response towards these, it's imperative to have adequate management in the Arctic. Thank you very much for your attention, and I will now open the floor to questions and constructive criticism.
Thank you, Elia. I will let Fernando start with the discussion.
Yes.
Thank you very much, Elia. It was a fascinating presentation. I think it's a very important topic. It's included in many European directives, and nobody knows really well what to do with it. How to measure it, how to incorporate it into impact assessments. So I think this kind of pioneering work is very important.
Yeah.
Having said that, I really would like you to explain me a little bit more. So at the end, you divided, something I grasped from your analysis, that you divided everything in terms of seasons.
Yes.
And in particular, I'm really interested, you didn't go back to those assessments of the ability of the method to detect the species.
In here?
Yeah, this one.
Okay, maybe this one? Okay.
So why did you divide
By
the-
seasons?
Yeah, the present substances according to season, and then you can move on and explain also a little bit how did you characterize the presence of vessels in the area?
Mm-hmm.
What's the importance of those coefficients that come out from the confusion
Yes.
matrix that
Yes.
make something very
Exciting.
confusing?
Yeah, oh, okay. Well, confusion matrix. Okay, so for the seasonality, I kind of entered this project with a lot of freedom. I could choose to analyze kind of how I wanted it. And I did have to make sure that I focused and I didn't lose myself, which sometimes occurred where I would focus on season. And then I thought, wow, it could be interesting to look at month. And then I was like, maybe I should look by week. And then I started realizing that if I spent too much time going down this road, because for each of these, I have 6 toll bands to look at. And so each time I did it, I had like 6 different things I had to run every time. And so at some point, I had to decide what was most important. And I did do a couple trials with the month, and it just kind of like, ooh, this month is significantly different than this one. This one is a little bit different. And so by looking at season, which I thought for the Arctic was going to be something, and something I read in literature that was going to be very important. And so that's why I decided to. Yes. Yes, go ahead.
I was going totally in the opposite direction.
Okay.
My question was, why are you dividing? Sometimes you just don't have data or presence.
In that sense.
You cannot detect any signal because probably the animal is not there.
You mean, yes, this is also why I.
Why not pulling together all the
Okay,
data?
okay,
Perhaps
yeah.
I should have.
Okay, no, that's me. I misunderstood. But that is, I think that is an interesting point and something I did learn, that it's going to not be there. And that's also maybe why I looked at presence versus absence. So I didn't show the graph just for time, but the toll bands were showing lower levels in presence, like lower sound levels in presence of vocalizations. And that kind of leads me to a question I don't have an answer to, which is, is this occurring because when we can hear their calls and their vocalizations, it's occurring in environments that are quieter, and it's just by the analysis that we could see it, or if there was something else that came into play, but yeah. I'm not sure if I answered your question.
Yeah,
okay.
More or less.
And moving to the vessel detecting. Would you like to see the confusion matrix?
No.
Okay.
No, if you can explain me, if that 73% is
yes.
good, bad.
I think it's good for. It's good enough to be used here. Okay. I think it should not be used. Maybe to make, but in a bigger setting or something more important, because I did notice these false positives that were coming up that were triggered. I didn't have time to look at maybe as many recordings as I could, and so for this, 73% is okay, but I was noticing that maybe the walrus knocks were triggering similar patterns to the vessel noise, and so I think it would be better to really go over more and more files to really see, like, is there one aspect that is triggering these false positives? And in that case, it would be good if we can figure out what it is. Because of all the boat signals, it has a good sensitivity. So anytime there's a boat, it does find it. So it is a pretty good.
What kind of real world data are you using to say, okay, this day I'm hitting a boat, there is a boat there?
I listened to the files.
Okay, have you considered perhaps complementing?
Yes. If I were to redo this, I would use AIS data. Like, I would have the shipping boats, and I would see if that really corresponds with what I'm getting as well. Yes, and many papers use it. I think you have to pay for it, but I'm not sure. No? Okay, then I tried to get it, but it didn't. I thought this would work fine for the setting. Okay. Well, thank you.
Nico?
Okay. Thank you very much for the presentation. It was really great. Quite easy to follow.
Okay. Good.
All the important results there. Congratulations.
Okay. Thanks.
I'm curious about how specific you are about fin whales.
How specific?
Yeah,
Yes.
I mean, are there other species of whales, like minke whales or others
Yes.
that could be there at the same season? But you assign that particular cause to fin whales.
Fin whales,
Why is
yes.
that?
It's because I didn't, so all of this data, oops, where is it? All this data right here, I didn't analyze. It was data that was already analyzed for this on the same recordings. So it's my colleagues who visually went through each recording and wrote down all the fin whale calls. And so it just hasn't been done for other species.
Okay.
But there are other species there, and that's also part of the variability. Maybe we can't notice it. Maybe there's other species influencing it as well.
Okay.
Yeah.
Another brief question I have was related with the negative. I don't remember now if it was negative or positive relation with amount of. I don't remember the variable. The amount of sound.
This one?
Yeah.
Yeah.
Against temperature. Negative.
Yes.
And why? Why do you think it's that?
I think it's because of the weather. So when you go into the colder season, so after October, November, December, this is where you have this increase of storms, rain, wind, and again, depending on the year, you might have some ice formation and then some ice break again that can happen, which I think is the case for October, November. And so I would say that's why we have the louder, the lower temperatures. Yeah.
That's all I have. Thank you very much.
Okay, thank you.
Welcome here.
Yeah, thanks. That's very good. And thank you for the presentation. Yeah, that's quite new for me. It's curious about several things. One is, can you evaluate or can you estimate abundance of species numbers or not only identification of different species, but apparently that's easy because they have different species, but also the number of individuals present. Can you?
Yes, I think it would more fall into something either using an algorithm, so through machine learning that would take the patterns and notice it, or it would be done by hand. But this isn't something that I looked at, and I don't think you can get abundance from the kind of analysis I did, which is more kind of number-based. I do know there's a bit of a, I'm not sure if it's a debate, but with the acoustic complexity index, that's just one index, but there's many others. And I know there is a bit of a discussion around, can it notice a difference between abundance and diversity? But I think the best way to get the number of individuals would be through manual analysis.
Calibrate with the visual observation or experimental fishing, for example? Could you calibrate sound with observations in the floor?
Yes, you could. I do think it's best because sometimes you can't see them, but they are vocalizing. And so you would do more of this listening and just writing down. Or again, since, for example, fin whales have very characteristic calls, so they're about 25 hertz and they're occurring very regular patterns. And so you can train an algorithm to detect that. And so you can just give it your recordings and it'll be there were this many individuals at this time. And it's a very exciting topic as well.
So you think that's good monitoring?
I do. I think it's great. I mean, you just
just
put your hydrophone, you leave, you can do other work, you come back, then you have to deal with the data. But yes, I think it's exciting.
The thing is, if you can calibrate abundance or even composition of species.
Yeah, definitely the composition of species. Yeah.
Not a large group, because waves, probably you can identify species, different species of waves.
Yeah.
But
yes.
perhaps for different species of fish or
it's starting.
Birds,
yeah, it is starting the fish. And again, it's just that you do have to do this calibration first with maybe a visual observation. You go and you're like, okay, there's only haddock here. Then they characterize the amount of calls. But for fish, it's difficult as well because sometimes one species will have 4 different types of calls and another will only have 2. So it's just a lot of training and gathering this data first and then being able to apply it to a certain area. But, yeah, it's very cool.
Very fun. Thank you very much.
Of course.
All right, thank you very much.
Okay.
I want to thank you, Elia, for your presentations, and we can give you a round of applause.
Okay, thank you.
And this also brings us to a close of this last morning session. So we will now have a lunch break, and we will resume at 3 p.m. at 1500 hours, as we say in the Army in the States. And so congratulations to all the presenters of this morning. And Joy, you've done a lot. Everybody's done a lot of good work, and you can be proud of yourself.
Okay.